streaming this?
We are now. That should click on at some point.
Okay, so I just have to give them a mic, right?
Turn the mic on and give it to them.
Okay, I'll try it. Yeah.
So here.
Sure, I'll try my best. So, I think. Welcome back, ladies and gentlemen. I hope everybody had a good lunch. So for our lightning talk student speaker, we have Mr. Kehendra Bhatrai. He is doing his PhD in University of Arkansas Department of Horticulture. He got his undergraduate from Tribhuvan University, Nepal, and finished his master's from Oregon State University Department of Horticulture. Today he will be speaking about genome-wide association analysis for downy mildew resistance in USDA Spanish germplasm. Please welcome Kehendra Bhatrai. Thank you.
Thank you. So my name is Kehendra Bhatrai. I work in the lab of Dr. Ainong Si. He's in the Department of Horticulture, and Dr. Jim Correll. He's in the Department of Plant Pathology at the University of Arkansas. The title of my talk is Genome-Wide Association Analysis against Downy Mildew in Spinach. The objective of my work is to evaluate and screen downy mildew resistance response in wide diversity of spinach germplasm that are maintained at USDA, and to conduct an association analysis based on the phenotyping effort from field on spinach. Spinach belong to Amaranthus family. There are some other important crops like quinoa and beet, sugar beet in Amaranthus family. It's a diploid crop. It's a dioecious crop, as you can see in the picture, female on left and male on the right. But there are some other forms of flowering in spinach, but like most are dioecious, and they are wind pollinated, so they are highly heterozygous. The genome size is almost like 1 kb, it's 980 MB, and the reference genome sequence is available, but they are still continuing to add and generate better genome sequence. In sometime next year they will release. The California group is working on it, and there is an increasing demand of spinach. Spinach, we basically know they are used as salad, but depending on places and different countries and different people, they use in different forms. We can see production. China, they produce more than many remaining 10 countries or more. US ranks second in production, but we can see on another chart here the production and head area, they are almost similar, stagnant for last 10 years. However, there is increase in price of spinach, so this is a good opportunity for growers. But there is a challenge of pathogen, downy mildew, which is very important in spinach because it causes around 20 to 30% of production loss. Downy mildew is caused by an oomycete. It's an obligate pathogen, Pornospora farinosa, often known as Pornospora effusa. The latent period is 6 to 8 days. So in greenhouse inoculation experiment, we will get the result after a week, and they are dispersed and disseminated by wind and rain. You can see the picture over here on real field situation. that the commercial cultivation practices. And then what we can see is like the patches of yellow, those are caused by the downy mildew, and they invest, the commercial producer, they invest a lot of money in removing those yellow leaf spinach from the field before the harvest. Otherwise, like, we will not buy spinach if we find, like, diseased leaf on the packet that we buy. So that's the important challenge. Here are the symptoms and signs of downy mildew. We can, like, in the field condition, they will show like gray to blue patches of spongy growth on the underside of the leaf. But in greenhouse experiment, we can easily observe the sporulation on leaves on both sides, upper and lower surface. Downy mildew are managed in the field using genetic resistance. And qualitative resistance has been explored widely, and several cultivars, commercial cultivars, are released that use like combination of one or multiple resistant gene in a single cultivar. Is a hybrid because they are dioecious and they can merge like at least 2 or more than 2 in a hybrid. We are emphasizing quantitative resistance, and we are evaluating field resistance on spinach, and that I will be like talking in these coming slides. Crop rotation is recommended, but it's not being practiced. It's difficult to practice because spinach, they grow spinach in California from Salinas Valley and then move down towards Yuma and provide like leafy spinach to consumers throughout the year. So they keep moving down, go south, like with from summer towards like winter, and then we can get spinach in the market. It's recommended to use drip irrigation, but it's still like I think like they use overhead irrigation. And most spinach, almost 50% of spinach, are organic based, for which chemical control is not acceptable. So breeding for disease resistance is prioritized. Downy mildew, there are many number of races, and the new races are evolving each year. In last 10 years, more than 10 races evolved. We can see on the picture over there, and using the differential cultivars, we can differentiate the different races, and the races are highly diverse. It's seen in the principal component analysis down on that picture. And in the field condition, the races or predominance of race differs among the location and the region and different season, and it basically depends on what kind of cultivars do you grow, like in your in the region, and what kind of cultivars they grow in nearby regions. Basically because different cultivars, commercial cultivars, uses different resistant gene, and so like they increase and decrease selection pressure. Utilization of host genetic resistance is most practical and economical and ecologically accepted method in breeding. In spinach, in commercial cultivars, major resistant genes have been explored and they have been used in releasing cultivar. However, new races are evolving each year. That's a great challenge. So we are trying to emphasize in screening for quantitative resistance and further utilizing those in breeding more durable cultivars, using both quantitative and qualitative gene in the same cultivar, if that is possible. This is how we screen downy mildew in the greenhouse. It takes about 6 or 7 days for us to complete the inoculation stages. We can do both, like whole plant inoculation as well as detached leaf assay to confirm resistance and susceptibility. It's easy to distinguish resistance and susceptibility. It's seen in that picture. However, it's difficult to quantify them. So we are trying to reach and identify the alleles that are resistant to downy mildew using two approaches: association mapping and QTL mapping. In association mapping, we'll use diverse panel and try to identify the associated allele to spinach downy mildew, and the tolerant line or more resistant line will be used to construct or create biparental population, and then we'll be studying QTL mapping on those in the future. The field screening effort that we use is we are screening more than 400 spinach lines. Almost 400 are USDA germplasm and 50 are commercial cultivars. They are collected from more than 35 countries, and we will be screening them in 2 locations, California, Salinas, and then Yuma, Arizona, for 2 years. In the first year, we just planted in 1 location in California and 1 location in Yuma, but this year we'll be planting in 2 locations in Salinas and 1 location in Yuma, and compile all the data, phenotypic data, and then use genetic data to conduct an association analysis. We scored them for disease severity on a scale of 0 to 100%. We took most severe leaf from each plant, and 15 to 20 plants were grown for each line, and we observed a wide range of disease response from Salinas in 2017. So I'll be just presenting about some preliminary data from Salinas. Next year, when we finish everything, we'll compile everything and then present all. Number of tolerant lines were identified, like you can, so I present here like top 10 resistant lines. These lines will be used in addition to others. These lines will be used to create a biparental population and conduct a QTL analysis in the future. And also these lines will be used to conduct association analysis and identify QTLs or alleles that are associated to downy mildew resistance in the field condition. Another aspect, genotyping. We submitted for whole genome resequencing of all those expressions that were evaluated in the 2 locations, so which will allow us to generate millions of SNPs. We'll use these SNPs or genotype data as well as the phenotype data to conduct an association analysis. And hopefully, I will get all this done by May or summer next year. However, like I'm presenting here like some association analysis based on GBS data that we already genotyped. The same, not all, but like many of those accessions, more than 300, were sequenced using genotyping by sequencing approach, and we have data for that. So I just use like those data to conduct an association analysis and identify the markers that are associated to the downy mildew resistance in spinach. But in next year, we'll be using the SNPs generated from whole genome resequencing data, and it would be like maybe like more closer to the real genomic regions. Yeah, and I would like to conclude that next year we'll finish our evaluations and conduct an association analysis, and then identify associated SNP markers. QTL mapping population will also be developed in coming years, and they will be used to identify markers from particular population, and this work should allow breeding for more durable resistant spinach materials in the future. And I would like to acknowledge my team, my professor, Dr. Anongse and Jim Correll, as well as my department head, Dr. Mackey, and my lab members, and the funding source, USDA, SCRI, and SCMP. Thank you. I'm ready for questions.
Hi. So you mentioned that you're going to resequence those 450 accessions at 10x coverage.
Right.
I guess, what are you basing that off of? Are you basing it off literature, your budget, or where is that coming from? And then, could you talk a little bit more about the downstream analysis from that, what you're going to do with that? to identify these SNPs?
So the question is, thank you for question. Question is, so where do you get money for sequencing, or?
No, how did you decide on 10x as your coverage depth instead of going higher or lower? Where did you make that decision?
Okay. So in practical, like what we are doing is we are sequencing 30 materials at 30x coverage. Those that we already know about, like those they are more tolerant or they are resistant and susceptible, and there's all those different cell lines. We are sequencing those at 30x coverage, and the remaining accessions, whatever like we are phenotyping, we are just like randomly sequencing at 10x coverage, so as to get like good SNPs, good number of SNPs, and then some other like downstream analysis as well. And these are submitted for sequencing, and hopefully we'll get result by in coming days.
Yeah, thank you.
Thank you.
More questions? Okay, I think let's thank Kahendra for his talk. Thank you.
Just as a quick announcement, if you guys brought a poster, we'll be taking those down right now. So if you could just come pick it up in the next few breaks, that'd be great. Our next speaker, though, is Dr. Matthew Blair. He received his PhD in plant breeding from Cornell University. In 2013, he joined the Department of Agricultural and Environmental Sciences at Tennessee State University, working as a plant breeder on new crops. So today, Dr. Blair will be talking mostly about his research on grain amaranth. So without further ado, I'll use that mic.
Okay. Thanks, Jay.
Good afternoon, everybody, and I put grain in parentheses because I know a lot of people are looking at weedy species of amaranth, so I wanted to give that as a background. But of course, where did we get the grain amaranths? They came from some of the wild species, and so I'll go over some of the phylogeny and domestication origins for amaranth. I'll also tell you why I was interested in amaranth. People always ask me, and my first job was at Rodale Research Center, and they had, in those days, and for about 2 decades, a small amaranth breeding program. So it was one of my first introductions to breeding. Then I moved on to legumes, and now I'm teaching mainly and working with marker-assisted selection and new marker types, especially for new crops. And my college is a college of agriculture. It's in a 1890s land grant, so it's a historically black university in Nashville, Tennessee. And as an MSI, we have some resources for plant science and animal science, but with a focus on biotechnology as a job-creating area of studies. That biotechnology is in plant, animal, and food sciences. So we're interested in things like new crops, like grain amaranths. We're also located in the middle of the southeast, as you can tell by the star in the middle of Tennessee, and in an area of climate change. So I know climate change is affecting the western states a lot, and also here in the Corn Belt, but down south we're on a borderline region where we'll have this gradient effect from -50 to +50 on the yields of corn, and I took that as an analogy for where amaranth can fit in and where it's possibly needed. So getting on to the project itself, this is a project funded by our Evans Allen, which is like catch money, and it's amaranth breeding program for Tennessee, and we've been very lucky at having a great climate to grow amaranth in. They really love humid weather, as most people can tell, and the grain amaranths are no different. They survive some of the droughts we have in midsummer, and there's a lot of diversity there. So here I've got a purple amaranth because there's talk of natural dyes from them. Jay won the People's Award, so at least having one of those, but I'll show some more pretty pictures of the crop. Amaranth as a grain has been around for 8,000 years, so this makes it one of the oldest domesticates, and it has a biomass that's comparable to that of maize, mainly because it's a C4 dicot species. After Spanish conquest, the Aztecs were prohibited from growing grain amaranths, and it was basically stopped as a ritual crop and also as a crop that was given in tribute to the Aztecs. And it really wasn't until the 1970s that there was renewed interest with some famous books, like the book by Noel Wiedmeyer on crops of the Andes. In fact, in India and China, it's taken off partly because of labor supply. A lot of the work around the agronomy of grain amaranths is still hand done, and we're seeing a resurgence of interest, or a new interest actually, in East Africa, where obviously there's a food deficit and grain amaranths can come into play. As far as classification, you've seen some of these pictures. I'll emphasize the genus at this point and the fact that amaranthus comes from the word amaranthus in Greek, never fading, or one that does not wither. So it's always been recognized as one of those fabulous plants that just survive a drought spell, survive a tough spell, and we see it often in our fields. It's also known in terms of the cultivated species as a pseudo-cereal, and this is mainly because the embryo is around the endosperm. The endosperm in the middle is surrounded by a circle, and in its relative, the quinopods, the quinoa, that circle will often fall off while you're cooking it. And then, of course, there are the 70 other species that are wild and weedy. And sure, we have a lot of weedy problems, but I emphasize there's still a majority of wild that are out there. Some of them are pretty interesting. Also that they're majority native to the Americas, and the rest are from the Old World. Their ecological importance is in that they're diverse and can adapt to a lot of settings, high altitudes, lowland, tropics, temperate. So many of them don't have photoperiod issues. Some of them are short day plants. They have high disease and insect resistance. This might be because people haven't found the diseases and insects to them yet. But they also are just very vigorous plants that grow very quickly, and you'll see some of the leaf sizes are increased compared to the weedy species on the cultivars. And along with C4 photosynthesis, they have a successful niche to fill, I believe. So on their origins, hybridus is considered one of the sort of origins for a lot of the genetics into the cultivated species, which include Cruentus, Hypochondriacus, and Caudatus is really a pretty separate offshoot from South America that has a close relationship with Quitensis, and the role of Powellii is still not very well defined. They're tall plants in the case of cultivars, and they have a single stem usually with less branching than in weedy amaranths. They have a good harvest index. They have a large range in flower colors, which makes them attractive and gives the idea of using them for dyes. But they have only small-sized cream-colored seed in the case of cultivars. As you know, the weedy species are black or brown seeded. And one of the amazing things about these seeds is that they pop. So as a popped product, they can create a lot of interesting desserts. They can be boiled like quinoa, and when they are, they're used as a weaning food. And in South Asia, I understand that they're one of the cheapest source of nutrients, cheaper than rice. So when poor people, lower castes, often have trouble buying protein sources, they can get it from amaranth. And it's, as I mentioned, of increasing importance in East Africa. This popping is interesting because I think it shows some of the ancient nature of the crop. Amaranth was probably domesticated before pottery was available. And if you think about it, some of the original mazes might also be originally popcorns, and some of the original beans at high altitudes were popped beans. So all throughout the Americas, people had this culture of growing grains that would quickly pop and basically on a hearth. and obviously they didn't need to boil them, so it was a quick way to have food. And the popping is almost silent. It's just small pop, 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 pop. So there are a lot of new ideas that come with new crops, and I thought, why not put it in a movie theater where you could, you know, add different flavors, and you could have silent amaranth going on right there, right next to your where you want your food. You know, you don't want to go all the way outside and get the food. So consumer traits that people look at is, how can amaranth be incorporated into breads, into chapatis, into pastas? And they've been successful at all that, and little by little they've been penetrating the market in the US. Once the market was established with some national production of amaranth, it actually started to bring imports from other countries, and grain amaranth is predominantly imported now to the US. But some of the original products with just popping and pure amaranth are nice. And in Mexico, you get a product called Alegría, or Joy. It's a snack bar made of amaranth and honey. Very good for you. Because of the nutritional aspects, amaranth is garnering this new foods market and whole foods, healthy foods market. And they're a source of a high protein grain that's gluten-free. So generally on those health bars, when they make those claims, they'll often use these ancient grains. It's also got some essential amino acids that aren't common in wheat flours. It's got high mineral content. It's also got a squalene that we were discussing as seed oil in a small amount, because it's not a very oily crop. And that can reduce cardiovascular diseases and also control hypertension, cholesterol, and some antioxidant status. So a lot of that is hypothetical, but it's worth thinking about amaranth as a diversification tool in the diet. There are 3 species, as I mentioned, that are cultivated. The caudatus generally has a weeping form, where the flower head goes and turns around and starts heading towards the ground. I think that might be having something to do with long growth cycle and the lodging that sometimes occurs in these plants. Acruentus is an upright form. It's in mid-elevation areas, and Hypochondriacus is a species with low elevation. So what's pictured here is a nice caudatus. The floral morphology, hearing some of the earlier talks, reminded me how much photosynthesis is going on in the seed head. So I think that's a strategy that's not only in grasses, but maybe translates here. There are issues about the level of C4 photosynthesis in many of the leaves of amaranth, from the cotyledonary leaves all the way to the next size, sort of immature leaves, mature leaves, and then finally these bracts that are surrounding many of the flowers. There's also a different ratio of female to male flowers, and all 3 of the cultivated species are monoecious, so they do have a tendency to self-pollinate to a surprising extent compared to some other species that have this mixture of male and female flowers within the same little branch or raceme of a panicle or a seed head. I call it a panicle for short. I know it's not really appropriate, but it's just easier than always referring to the large seed head that we have in amaranth. We do have leaves that will come out directly from the seed head and true leaves, true large size leaves. There's infrequent occurrence of male sterility, and so far there's no hybrid breeding, and crossing tends to be difficult because of 2 things I'll mention here, which are hand emasculation. If you thought the spikelets in sorghum were bad, try amaranth. As a younger version of myself, I was able to do that, but my eyesight is gone now for hand emasculation. So recently, Stetter et al. Came up with a hot water treatment of the females, where I don't know if you can tell, but they're dipping in the middle panel. They're dipping the females, which in this case are green, to show the hybrids by crossing them with red males, and the red males are there for subsequently placing the two seed heads together or flower heads together and getting natural pollination. Natural pollination by itself, you'd think with a potentially outcrossing species would be a good way to identify hybrids, and really there's not much natural pollination. It's a sort of lower percent range in many of the cultivars. So maybe this was selected for by people to have more stable lines when they were growing cultivated amaranths, because they tend to be very different than the weedy species, which is one other thing I was reminded to tell you, which was don't be worried about the cultivated amaranths. They're not going to kill you. They're not going to kill agriculture in the U.S. I think I'll skip this, but growth chamber experiments modifying day length can get Amaranthus caudatus to flower, and then that cross can be made. So this photo period I find equivalent to stress responses. So amaranth plants are very plastic, very able to adapt to many environments, even the cultivars. So like some of the weedy species, if they're only given a 96 well tray amount of soil, they will grow correspondingly to only 1.5 feet in height. But if they're given a full soil site, they'll grow like the ones that I'll show you in a couple of minutes. And this high level of plasticity allows them to flower at early intervals, and as a result, they've been suggested as a model species that can have 6 generations per year, but they also can adapt to drought conditions and low nutrient soils. So we find that many of the cultivated amaranths do need relatively rich soils. That's how they're grown in their homeland. So those varietal differences in flowering, you can just start to see here that these are predominantly Hypochondriacus and Cruentus amaranths, and you get some full flowering in young plants, some incipient flowering, and plants that remain vegetative when they've been grown in the spring greenhouse to be transplanted outside. And all of them can do pretty well outside. Even the ones that flower early will send up side shoots, and they'll flower like a normal vigorous plant. Our materials and methods for the experiments we've done so far have always consisted in amaranth germplasm screening with a set of accessions from the Seed Saver Exchange. Not sure how many people are familiar with that. They're based out of Decorah, Iowa, and their landrace varieties and breeding lines saved from the Rodale program were never tested in the South, so we wanted to do that as a first step. We did that from 2015 and 16, and then started the USDA core collection, which is set up by David Brenner, who many of you know, who've worked in amaranth weedy species, and he's based at USDA in Ames, Iowa. There we use 260 accessions. In our Seed Savers Exchange, we just do, because we're transplanting, randomized complete block design, 2 reps. We don't fertilize at all. We don't use any pesticides. We use 3-meter long rows. We use 50-inch spacing between rows, and we greenhouse plant in jiffy pots and then field plant in June when it's gotten warm enough to reliably have the amaranth growing. Now we've done 2 methods. We've planted by transplanting, and we've planted also by direct seeding. And our direct seeding actually works just as well as long as we hit warm soils. If we plant in soils that are too cold, we won't get good amaranth germination, and instead we'll have lamb's quarters and other chenopods coming up, weedy species, and then we get a mix of some of the weed amaranths and some of the cultivated amaranth that we're planning to grow. So soil preparation is very important for grain amaranths. To give you an idea how long it takes to harvest, we usually wait until September, and by then we've had a dry spell and the plants are starting to dry down, at least those that are adapted. And that's really a 5-month growing period. So you can tell that some of these amaranths take a long time, even in our long days, summer environments. But there are a number of them that will be ready in August if you plant in June, and some that will be ready in November. So there's a range there that you can play with in terms of months to maturity. We still have to harvest and dry our seed heads in a hoop house for about 1 week before we're confident that we're not growing molds and other things on our panicle. And then we do a biomass of panicle, biomass of stem, and a weight of grain. We also take morphological markers with the idea that in GWAS studies we'll be able to evaluate all these as maybe simple loci. And we do find that there's some phenotypic plasticity for pigmentation and some of the other traits that you would think would be more stable in amaranth. So again, something about the amaranth genome allows for a lot of these things to be, gene expression to be changed. This is a scan of all the seeds of the seed saver collection, and it shows you that it ranges from the black seeded types, which tend to be vegetable species, variety, and then some gold and some cream and some white seeded types. And you don't see much in the way of seed size differences. So, you know, if you look closely, it's easier to see with the lighter colors, but generally the gold is the same color as the cream. This is a transplants being ready for the greenhouse. Sometimes they'll be transplanted into the Jiffy pods. Sometimes they'll be direct seeded, and then we usually try to get plants up to about this height before we take them out to the field because they survive better. You can see that you can wait a little bit too long or have some that react to photoperiod and will flower on you. This is the actual plants out in the field, and like I said, we would do 3-row plots and 2 replicates. That variability within the plot in the first set of 3 actually came with the accession that we got from USDA. So there is still sort of a pigmentation purpley green to greenish purple mixed together. We always plant legumes around our borders, and that sort of gives us an idea of how fast the plants are growing. We also use corn on the side of our fields to just compare a C4 with a C4. And this also allows us to notice any lack of uniformity in the soil up and down the rows. And you can see the plot of three rows and compare that height to the legumes. And I'll just show a bunch of pictures showing when they're uniform, which in most cases they were, with accessions brought from USDA. And as a result, I mean, from Seed Savers Exchange, most were uniform. And we were able to whittle down over the course of the last 4 years to get a few entries that could be potential recommended varieties for people that have expressed an interest. Now, since I'm in Nashville, Tennessee, the people that have expressed interest right away have been the whiskey distillers, okay, the beer makers in the microbreweries, okay, and some of the dyes, and then some of the home bakers and vegetable gardeners, that sort of thing. But I was really amazed when the distillers came in and said, I'm sure the amaranth is going to give a good taste, because they had already tried quinoa and they wanted to try amaranth. In the case of beer, they can actually use the bitterness of the leaves as a hop substitute and make beer out of it. So we wanted to have a list of 4 best entries, now with direct seeding rather than transplanting in strip trials, and I'll show you those pictures at the very end of this talk. I think I'll skip over this for Seed Savers Exchange, but they're just an heirloom seed place, and if you're interested in any of the varieties I'll mention here, you can contact them and order them online, basically. You have to agree to exchange, and also they do have an MTA now. It turns out that many of the varieties that worked well, the 4 that we selected for our strip trials, were originally bred in Pennsylvania under the Rodell farm, and they come with the R designation, or this K112, which was from a breeder set up in Rodell named Kaufman. And Kaufman really wrote the book on a lot of the amaranths when they first were coming out. So he was always asked to write the review papers and things like that. And I had the chance to work with him and his right-hand field person. So these are the best varieties, and pretty much they're all from Rodell. On the USDA collection, we tried a number of ways, and I was lucky enough to go to the greenhouse over lunch and see that people are growing amaranths as in pots very densely. And you can actually grow them under those stress conditions and transplant them to the field. So that's one thing that we did, and we had a row by row comparison of transplanted seeds from pots that were ripped out and ones that had the jiffy pot of soil next to it, and we wanted to see whether they did better once transplanted. There was no difference whatsoever. So amaranth is one of those plants that even in Mexico in the traditional planting systems is ripped out of the soil and planted just on a good rainy day, you know, or in a furrow that's been irrigated with mountain water in the case of Mexico. And we've had high school interns involved in this program. So this is growing them in jiffy pots en masse for the USDA, and we set them out in 2 rows of 36 plants each, with rows at 4 feet centers based on tractor width and tire width. And that was because we used a transplanter to make the holes that we were going to use at an even spacing. And there was 6-inch spacing between plants. So that was a big trial, but one that we were happy to show because we hosted the Amaranth meeting in the US, and a lot of the Amaranth people came in and saw that field that year. So it was a nice time to see the diversity of species, and also for the people that were weed scientists not to be as scared of the genus as some of the farmers that we heard about in Missouri. So the USDA core collection was planted in a similar way. It was planted just a little bit later than we normally planted our seed savers, and it was also started a little bit earlier than the ones that we had done for seed savers. And there was a lot of variability in terms of uneven harvest. So we also had to dry and get the seed heads dried before we process them. And here you can see the first trial, which was for morphological traits, and you can get a general idea of our field and how they're growing compared to corn in the background. Because of the transplant, they weren't growing quite as fast as corn. But now when we do direct seeding, they grow almost as fast as corn does if planted at the same time. So I'll just show you a bunch of pictures showing you differential traits, weird dwarfing types, some potential virus symptoms, purple leaves as a common trait, stem colors of difference, and the amount of flange that you get. The curving of the roots here actually came from the initial jiffy pot where they had started to grow in a circle. Then flower color and flower branching, so either a compact panicle, an early flowering, a drooping panicle, continuous flowering with the side shoots producing seed, purple leaf with purple panicle, red, green leaves tended to have pink panicles. Pink panicles are pretty common with both green and red leaves. Orange is not a very common color, but is found in a certain number of cultivars from Mexico, and also breeding lines from the Rodell program, and then just a yellow-green panicle. These tended to be the most stable in terms of just stem, and they were more prevalent, the green ones. We also thought that they would be good because if we used them and there was any weed contamination, we'd generally notice the colors brought in from the weeds. Compact panicles and then open panicle, and there's something that David Brenner even calls a determinate panicle, where there's sort of a globular shape to the groups of flowers, the flower clusters. And you can see the leaves growing right into the flowers themselves. Okay, this shows variability in plant heights, where the average was 1.2 meters in height. The range was from dwarf to 2.47, 2.5 meters. Seed yields, obviously, when you're looking at a initial collection and selecting parents, a lot of them are toss-outs, and so many of them would yield very low, below 54 or 50 grams per plant. but some of the best reached up to 540 grams per plant in these separate conditions. Now that changes once you put the plants in direct seeding and they're competing within row amongst each other. The panicle biomass was up to, was average of half a kilo, and the range was 27 to 2 kgs in grams per plant. So you can imagine very large plants with very large seed heads, but that includes the stem value up to where the flowers start and where the seed actually pollinate, where the seed is produced. And then the percent of seed weight in panicle was calculated to be an average 21% with a range that was very large, depending on whether the plants, the seed heads, dried down quickly or whether they dried down slowly. This could be useful for selection of a dye source with high panicle biomass but with less successful pollination. That might range from environment to environment. So our present research focus is in genetic studies of several new crops, and the pseudo-cereals, mainly amaranth, is one that I work with, along with subtropical legumes that are close by. And my area of interest is in drought and heat tolerance, but we have a way to go, I think, for the amaranth work in that area, and then training of students. So with molecular markers, we found that this was a good way to introduce students to diversity. And we started with genotyping with SNP markers that have been developed for amaranth. There's a range of CAS markers that are easy to use. The SSRs I find are a little bit limited compared to what I've worked with in other crops. But GBS for us in our hands has been very successful as a genotyping method, so it's our method of choice for association mapping and GWAS studies. So all the range of previous markers had been used, but I'll point out the last two with papers from a group in Utah at Brigham Young that have developed simple sequence repeats and single nucleotide polymorphisms where they converted a lot to biallelic Cas markers. We get our DNA in a sort of funny way, so I thought I'd show how we do that. Especially for GBS, we like to use these magenta boxes with agar media, and when we have that, we can get a lot of fresh young tissue and high quality DNA. So we've never tried any field collected DNA. This shows you one of the species starting to germinate and the reaction, and then a whole group of species ready to harvest for any type of fast DNA kit. Any of those prep methods are good. The SSR amplifications are relatively easy. We've only tried 50 markers. What we find is that they tend to stutter a lot, and so they're difficult to read on gels. So we've gone to SNPs with Cas markers, and we had a group of 45 at a time synthesized, and we've gone with what the company LGC recommends. They're the basis for Cas genotyping assay, and we have some of the equipment for that, which I'll show in the next slide. It also uses a very small amount of DNA, the 10 microliters, and you can change the volumes of the reactions from 5 to 10 from 10 microliters to 5 microliters very easily, and still get a good result. So we actually use some of the robotics for both DNA extraction and then also for dispensing, and then we'll use a plate sealing machine called a cube, and we'll use a version of this washing machine-like object, and that's the old method of PCR where you're using water baths, and it's quite efficient because you can get short run time, and you can also get good separation of your alleles by the allele binding of the specific primers. There's a 3-primer set that works in every Cas reaction. And the data reading is fast because there's no gel involved, it's just a fluorometer. spectrophotometer. And we were exposed to this and thought that it would be interesting because we understand that a lot of the companies use cast markers still because they're so flexible. So as a training platform, we wanted to have this for our students. This just gives some of the cast protocol for SNP markers, and the software is quite good for it, and it's quite flexible. Genotyping by sequencing has emerged as a rapid and direct study of sequencing, and the only problems that we've had is providers for genotyping by sequencing. So we plan to do a more in-house GBS, and using some of the new recommendations from Illumina on targeted sequencing. So I think that'll be useful for a small crop that can't afford some of the resequencing experiments. But our GBS has been based always on good DNA quality, checked by a UV light evaluation and a pre-digestion test. And that was important for generating a large number of SNP loci. This is the distribution of SNP loci that we could use in our diversity experiments, and we found that they were well distributed on 16 of the largest sequence scaffolds represented in the phytosome database, and they were all in accordance with Jeff Mond's recent publication, and he was kind enough to share with us the version 2.1 of the genome. So with that good coverage, and you can see that it looks like there's some sort of centromeric regions that are not covered by general GBS, but most are. We were able to find overlaps of the SNP loci in the different species. We included 2 weedy species, Retroflexus and Powell amaranth, and you can see those on the outer edges of this Venn diagram. And in the Venn diagram, you see the little cross, the square, and the cross, the circle, and the spoke wheel, and each one of those represents some of the cultivated species, with Quitensis being a wild relative that's closely related to Caudatus. That's why you see such a nice overlap between the circle and the spoke, if I'm explaining that right. The barbell in the middle is A. cruentus, and it gives a number of unique SNPs, which was close to 6,000 for A. cruentus, that distinguish it from all the other species and within the species itself. So GBS has been a useful method for cross-comparing other species, both in our lab and also in a German lab that we saw has done GBS work across the whole genus and for phylogeny. This is our dendrogram of 6 amaranth species, and you can see the separate branch that takes the dioecious weedy species is annotated in brown, with open brown circles being the Retroflexus and closed circles being the Powell. And then you can see all the other ones, except for the Quitensis, which are indicated in light blue open circles, are relatives of the cultivars. And so we've separated wild dioecious species from cultivated monoecious species. That makes sense because they're in different parts of the subgenus. Among cultivars, we've separated basically the Caudatus and the Quitensis together as South American origin species from Central American origin species being Cruentus and Hypochondriacus. And then in these side B and C, we've shown that there's an association for seed color and the population structure being in B, and origin of each accession being in B for South America, Central America, green versus blue, and then the seed colors are the actual seed colors associated with the accession. So we have some red types or reddish pinkish types, and those occur mainly in the Caudatus, and then we'll have cream distributed in both clades. This is a principal component analysis. It just shows again the close proximity of the Quitenses and the Caudadas, all being at the right hand of the diagram, and then a wider range of diversity for the two Central American groups, the Curentis and the Hypochondriacus, being blue and red on the left side. This is a population structure with the same color coding, and we have at different levels of population substructure more and more separation of those that are Mesoamerican from those that are South American in terms of accessions, and some small grouping within the Hypochondriacus that might show some subpopulation structure. This was all necessary for doing GWAS because we need to know the population structure and the kinship matrix to evaluate any associations that we might find. In previous GBS studies, they did a phylogeny of 35 species of the 50 to 70 or so in the genus, and they found this grouping under the subgenera Acnida, Albersia, and Amaranthus. And Amaranthus holds all the cultivated ones plus the hybridus. So the hybrid, Amaranthus hybridus, can also be a progenitor of some of the cultivated amaranths, or maybe have been introgressed. And but there's still sort of an outgrouping of them compared to the cultivars. In the Acnida, you have the main amaranthuses that you're worried about, including Palmer and tuberculata. This is from their paper, the paper of Stetter et al. In 2017, with an interesting marking. I don't know if people have seen this before, but the way they've drawn their phylogeny is with color-coded bars or lines to each species and a little picture of each species. So generally the ones that are in yellow to green have the intermediate level of genome size, and from GBS they discovered this. Those with red or orange have small genomes, and those include Palmer amaranth, spiny amaranth, and arenicola. And then, if I go to the next slide, you'll see the group that has potential polyploidy, and those include some vegetable species, like tricolor, which is in the group Albersia, and some other species, viridis, I think is sometimes used as a vegetable. And then another part of the Acnida have diverged from that group, it seems, and so there's a little controversy about the subgenus. But they include Australis, tuberculatus, and Floridanus, and those seem to be doubled in the amount of DNA that they have. It hasn't been confirmed cytogenetically, but it seems like they have a high amount of DNA. So this just shows that and highlights in blue boxes those species that are potentially polyploid. And this is their understanding of diversity at the moment. And we were interested because we wanted to make some crosses between our cultivated species and some, potentially weedy species that to understand the basis of weediness. And we found that really within the group of cultivars, there's only hybridus, and pretty far away are the other wild and weedy species, except for Puelli and Retroflexus. So a revised origin or idea of cultivated amaranth is that A. hybridus really gave rise to all the Central American and contributed to the A. caudatus together with A. quitensis. And so that's at least what we know so far, and I think it's a way to imagine how to do population and population genetics work in the cultivated amaranths. So going back to today's topic, which was obviously agricultural solutions for a rapidly changing world, we've got the potential for new crop development from the grain amaranths. There's 3 species that are already available. They're being taken up in East Africa, which has some similarities to Mexican homeland for these species, especially the Curentis and Hypochondriacus. And they have wide adaptability. Now, one issue that I've tried to go over and over again is architecture. There's lots of variability, but really there's been a discussion of what type of plant will yield best. An upright single step, or a multi-branched, maybe decumbent growth habit, or a candelabra shape. And there'll be a few pictures at the end where Iowa State has developed a dwarf amaranth that basically has a candelabra shape. So what would allow for mechanical harvesting? What would reduce harvestable harvest index or total yield? And that would be important in areas of food security and when you're doing hand harvesting. A final issue is larger seededness, and there's very little diversity in grain amaranths for seed size. That potentially polyploid source, A. australis, which is actually from the southern part of the US, has a slightly larger seed, and so some idea of crossing with that, but it seems unlikely to do, and I'll show you why. This was our A. australis, which I call tree amaranth, in our field, and if you ever want to scare a farmer, show them a 25-foot amaranth plant, okay? This was looking up. I mean, I couldn't even get the whole plant in. And then this is another large seeded species that's more of a swamp plant. So I think we'd give up some drought tolerance by crossing into that. So that just gives you some funny pictures of and in the background of that one, you can see the regular amaranths with their large red seed heads. So our amaranth US studies will be basically with our GBS markers, currently about 10,000 for that set of species, and good for identifying easy to measure traits, maybe morphological traits, plant architecture, and height especially. And then collaborations on biochemical traits, stomatal density, water use efficiency, or photosynthetic efficiency parameters. And this would include the dyes that is of interest here. Seed color and size will be difficult to measure and evaluate given the low diversity, but it may identify major loci for domestication in the Americas, especially the seed color, because it seemed to be very important to get to a cream color. And it seems to be a little bit larger, the cream seeded. In herbicide tolerance, amaranths are well known for developing herbicide tolerance, both in the glyphosate resistant, and so it'd be neat to know how much malleability the genome has in terms of EPSP and how it compares to Palmer amaranth. But Palmer amaranth looks to be a smaller genome and especially more rearranged for some of its resistance characteristics. So the current role of cultivated amaranth might be more important as a way to control weedy amaranths. My experience shows that you get fewer spiny amaranths when you grow a big field of grain amaranths. And that seems to make sense because A, you get cultivated volunteers coming up at the same time as the weeds, and then competing for that amount of space. You go through and you cultivate once, and you can get rid of, if you've planted right, you can get rid of a lot of the weedy amaranths. So I think that approach that one of our speakers gave about a new way of controlling weeds is right on. And I'll just show that to you in this recent picture. This is our 2018 planting, which has been wet spring, dry summer, and then maybe hurricanes coming along. So you can see on our right the effective grain amaranth rotation, where there's less spiny amaranth in a soybean field that's been filled in as a rotation crop to give nitrogen to our organic grain amaranths next year. We've got some sweet corn off to the left, and you can see that it hasn't done very well because of the drought, and it was planted at the same time as the soy and the amaranth. And you can see grain amaranth in terms of biomass being well, twice the size of soybean or more. On the other side of that same planting, and these are seed saver exchange varieties, we have cowpea. Cowpea is a legume that tends to sprawl a lot, but we have grain amaranth growing in pretty good uniformity we come and rogue out any of the little reds that you see or pinks in our green varieties, and those tend to be F1 hybrids, usually between accessions. But this is actually field saved seed grown to field in direct seeding, sort of what a farmer would do. And if we get any crossing, well, it's population development, okay? We were thinking about a real population. We were able to get up to F5, F4 by crossing one accession that Pat Trenell donated to the collection at USDA, and it has some herbicide tolerance and is from hybridus, and was collected in, is that near here, Edgar County, Illinois? Not too far. And we crossed it with Plainsman. We didn't cross it with Plainsman, it was crossed, I believe, here, and in collaboration with David Brenner, we have the cross and advanced it from the F1 seed. The F1 seed was selected with Pursuit as an herbicide treatment. And then we'd grow this in a greenhouse with individual pots, and we'd use glycine bags to cover up some of the flowers so that we get self-pollination on those and develop our rills. We don't get a lot of seed under the glycine bags, so I liked an idea that I heard earlier where you just grow the plants very small and keep them covered from the sides. This is amaranth population at early stages of development, and next steps would be to get it into a QTL evaluation scheme that helps the GWAS results. So I just wanted to end by encouraging developing country and vegetable or community gardening uses of amaranths. This is experience I've had with the ethnobotanical program at Vanderbilt, which is right next to us, and they have really taken to growing amaranth together with beans, maize, and pumpkins. Each seem to have an ecological advantage. So if you consider the three sisters as being the maize, the beans, and the pumpkins, maybe you should consider amaranth as a fourth potential sister. It's also a crop that, because of its drought tolerance, could be grown where pearl millets, sorghums are grown in the Guinea Savannah region of Africa. And since they're now pushing soybean into that region, it could be something that could alternate with nitrogen-fixing crops like cowpea, which is traditional there, and sweet potatoes. So you could think of systems for farmers that would be holistic and that would include amaranth as an extra food source. And amaranth is great as a vegetable due to its high drought tolerance and ability to regrow when it's cut. And pretty much any young amaranth can be used as a vegetable, as long as it doesn't have spines this long. Okay. So I wanted to end with this picture of breeding lines from Iowa State that are semi-dwarf, and you can compare that. This was a year ago when we had better rains and the corn grew better, so it's about half the size of corn. So that gives you an idea of what potential plasticity in the crop can do. These were a comparison in the same season where the same person, namely Dr. Dafeng Ding, was right next to traditional amaranths and semi-dwarf amaranths. So those might be useful and are available from David Brenner. We've done promotional brochures just to get local farmers interested, both in the leafy amaranths and the grain amaranths. And I'd like to acknowledge undergrads that have helped, and bachelor student Matthew Edwards doing the DNA extractions, Ranjita doing the GBS together with Shimbo. She started with her CASP assays. And Damba Yahaya, who's from West Africa, and has said, I'm going to take some of your USDA accessions to my country. And so I hope that they have it traditionally in West Africa as a vegetable, and I hope it grows well. And the funding has been with fellowships and with Evans Allen from USDA. We have yearly meetings for the Amaranth Institute, and please visit, easy to remember, www.amaranthinstitute.org. And with that, thank you very much. Toki?
So, are there ever any issues with shattering in the cultivated things, or is it fully non-shattering?
No, for sure. It's sort of a semi-domesticate in that sense that it will shatter quite a lot. And someone was asking me, Jay was asking me about birds also landing on the heads and sort of moving them around. Now, I don't think that's as much of a problem in Mexico, and we haven't seen it where there's other food sources. They don't like the amaranths per se, more than, say, oily crops. But as a problem, I think shattering is probably one of those, and it's hard to address because it's those clusters of female flowers with a few male flowers, and they tend to dry down at different times and things like that.
Just one other one. Do you ever run into any pre-harvest sprouting?
No, that absolutely not. So that we're lucky in. I don't know if anybody else has, but amaranth will mold before it starts sprouting. So, and it'll drop before it does that. So one way that a farmer could grow it here is a little bit like soybean, where they use the freeze to dry down the plant and then harvest. So one of our goals would be to fit into a cycle where you'd have, say, a winter crop in a cover cropping situation, and then plant amaranth, either no-till or just a little strip till to germinate. And I was thinking canola might be a good one in the south because canola doesn't come out until late May. And so this is a plant that loves to grow in June, July, August, and then zap, October 30th, you know, we get a freeze and be ready to go in and harvest. And you lose some, but that's where I think you get your weed control of spiny amaranth, is because there's so much on the ground that it tends to cover in the same places, and you get good competition with the other amaranths for sure.
So what's the feasibility of mechanical harvesting? Is that, I mean, is it economically feasible at all, or,
Yeah.
or what needs to be done?
It was done with one variety, the one that looks a little bit like this last picture, Plainsman, and it was grown out on the out in Nebraska, and it was basically a dryland crop, and they would allow it to dry with the season and only put a little bit of irrigation on, and then they would harvest it sort of as a semi-dwarf, like the ones I showed, except it was all based on that phenotypic plasticity. So if they got a late rain, the plant would grow too tall. So that was a problem, but they used some sort of combine, I'm not sure which one, that would get the small seed.
Would there be any risk of this kind of a crop going weedy and becoming something where you would not be able to keep it from taking over?
Right. That's the other question that people ask, and I think the example of Rodale is that no, it never will become a weedy, and the example from the phylogenies is that it probably won't cross too far out of its subgenus. I mean, or within its subgenus. So it's probably not going to become the next water hemp hybrid, you know, or cross with spiny or anything like that. So maybe there are some crosses with retroflexus, natural crosses, and with hybridism. I don't know if Pat here has experience with that, but I don't believe it'll be a weed, especially on its own, because it also tends to be single stem, and so that's very easy to control, you know, mechanically. Whereas when you have the branching down low and you mechanically go through the field, like on spinies will break off, and then they'll, you know, make a whole new shoot. You can cut off half of the root, and the other half of the root grows. So I don't think so. Is there one lady in the back? But then.
Are there any other questions?
Oh, okay.
Okay. All right, well, let's thank Dr. Blair again.
Thanks a lot. Okay, so I'm going to introduce our next speaker today. It's my pleasure to introduce Dr. Gene Giacomelli. Dr. Giacomelli is a professor of biosystems engineering and an adjunct professor in the School of Plant Sciences at the University of Arizona in Tucson. He earned his bachelor's degree in horticultural sciences and biological engineering from Rutgers in 1977, his master's in agriculture engineering from UC Davis in 1980, and his PhD in horticultural engineering from Rutgers in 1983. Dr. Giacomelli has designed, constructed, and operated various types of greenhouses using both hydroponic and aeroponic systems for lettuce, tomato, strawberry, and other crops. He has collaborated internationally for nearly 70 years in research, education, and the development of sustainable methods of growing food to feed the world, as well as designing food production systems. The title of his talk today is Why Controlled Environment Agriculture is Propelling the Change for All Worlds. Please join me in welcoming Dr. Giacomelli.
Okay, thank you very much. I think I can safely say you're in store for something completely different than what we've heard all day long. So, but first, let me thank the planning committee, particularly those who stayed with me and were trusted I could bring honor and some respect to my aspects and interest in controlled environments to this group. So I look forward to that opportunity. So to say that you'll have something completely different, I will start by showing something that you recognize, and then we'll move onward from there. All right, let's be realistic here. I think you recognize this plant, and I just said it's somewhere in Illinois. I don't know, I got it off the internet. But here was an opportunity that I get to appreciate what it's like to be among 1 million corn plants. And yes, you can hear them grow. What we have here is the controlled environment agricultural equivalent of those 1 million corn plants. Here we have about 13,000 plants per acre in a greenhouse growing tomatoes. This is in Arizona, and some of the numbers, 220 million pounds in this facility of 270 acres per year. It's a lot of tomatoes, it's a lot of water. At one point in time, this Eurofresh company had about 25% of the fresh market tomato industry in the US in the early 1990s before others caught up, and particularly Mexico. But here's one of their first, this was their 45-acre block, airplane view of the greenhouses, glass greenhouse. And then they moved on. You can see number 1 is way up in the top. Group 2, 3, 4, 5, and 6, so they got up to 272 acres. Because there was a market demand, they got so good at building, they had their own construction company. And during the early 2000s, they expanded from about 50 acres through this 272. Field production agriculture has nothing to worry about. The greenhouse industry will add a supplement to what can be done. I would venture to say the most productive, the most capable to provide inexpensive food that the world has ever seen, because of breeders, because of nutrition, because of irrigation, water use practices, and because of engineering mechanization. Now there's this term precision agriculture, and we see that this offers more information to the grower about every 10 square meters of crop throughout his or her farm to make decisions on the go about fertilization, about herbicide application. The historical information about the crop grown in that field can be utilized from year to year. I, though, stop at a point and say there's limitations to precision agriculture in part because you lose the option for control. You're at the whim of nature. And I put this picture up, and you see the date there. Can you see that, June 26th? What's wrong with this picture? What's wrong with this picture? It's not so wrong, but coming from an engineering point of view of a controlled environment, that date and that corn, what's wrong? Any ideas? No, I stumped you already. No, come on. What's significant about June 21st? First day of summer, longest amount of sunlight per year, day of the year, and therefore ever after until the next year it's shorter. Yet you have that corn knee-high by the 4th of July, is that what I understand? Has a long way to go. It needs a lot more photons of light, and it's growing in a situation where the days are getting shorter and shorter. Now, nature's figured it out. An engineer might say that's not being very efficient, but nature's got it figured out. One thing we realize is that the weather is also warming, and that's very important for the formation and the development of the corn. So we'll give nature the respect on that one. But here's what we can do in controlled precision agriculture. Grow a crop, in this case not corn, lettuce, but other crops, like you saw the tomatoes, that can be produced any day of the year, providing the environment that it needs to grow, and target that and have a dependable supply of it when you need it, predictable supply of it when you need it. But I bring you back to this picture here because this is a project that we've been involved with for a couple of years in growing this funny looking tall crop that you see here in a greenhouse for production and generation of seed corn. And this effort has led, because of the reduced amount of water, the number of replications per year, up to 4 crops per year in the greenhouse. Christmas time, being in the greenhouse and smelling the corn pollen like you could in July in the open field is somewhat amazing. I ask you to take that opportunity sometime. And it brought Monsanto and now Bayer to the point where they're building a $100 million 7-acre facility, greenhouse facility, to use this technology that we helped develop using hydroponics. No soil, no earth. There's still soil root zone. Anyway, with that as an introduction, I could go further about who am I, and maybe there was enough detail already. But I've been around for a while, not quite 70 years, but certainly 20 in my former life at Rutgers University, also in controlled environment agriculture, and the last 18 at the University of Arizona in the Controlled Environment Agriculture Center, always working in greenhouses and focused on education, research, outreach, and development of business alternatives. The division of the Controlled Environment Ag Center, and I was the director until March of this year, and now I'm free at last to do a lot more exciting things, a lot less administrative work, and have 3 new graduate students. I'm very excited about that. But these are the people that are working to develop these systems that are, as it says here, environmentally, economically and socially sustainable option, not replacement for field production agriculture. And we continue with educational programs that take the basic sciences and apply them in using engineering technology, math, and solve practical problems that face the industry in a controlled environment. And there are many of them. But we've come a long way, and some examples that I'll be going through: applications from the greenhouse tomato industry to growing in the earth in a high tunnel, to grow rooms, and and vertical farms, as they're called now, and even food and space. I should mention that I was initially asked to speak about the Mars Lunar Greenhouse. And I was glad to accept that and realize that there's not many other people walking around on this planet that can talk about Mars lunar greenhouses. So I don't know what that means, but I'm glad to be here anyway. The point is, I wanted to make a bigger picture around that. And the fact that every system you see here is nothing more than a controlled environment with the expectation to provide the plant what it needs so that it provides us what we expect from it, whether it's food or whether it's a phytochemical or whether it's just biomass. So we have these many opportunities. A big part of it is education, and I saw that in several presentations today already, is to get the growers to buy into it or to get those who have some experience to begin using the science and engineering that's behind controlled environment to their advantage. So we do a lot of short courses and tours and have many visitors. These people that work at the Controlled Environment Ag Center, faculty from the sciences, from engineering, staff, students, volunteers, people from industry are focused on these topics here, from energy through capital resources, including labor, nutrients, and water. Labor is one that we forget about a lot, and that's a big part of what we have to keep in mind. If you understand that CEA systems can be utilized and produce a crop or a product anywhere at any time, then you need hardware systems, and the examples we'll look to: greenhouses, growth rooms, so-called plant factories, and most recently the term that's come is vertical farm, which the plant never sees the sunlight of day, but only is produced with electrical lighting. So when I put here anywhere, and this brings me back full circle, why not have food production systems for space? And the term, and I believe you had a speaker here, Joya Massa, in the past, who spoke about her work at NASA, and this is NASA-related work and supported work for bioregenerative life support system, where I'll show you the Mars Lunar Greenhouse, whose purpose is much less for food production than for two other much more critical things in life that you will not survive without more quickly than food. Right? Oxygen is the first one, water is the second one, and then food, calories, and nutrition and whatever. And then we'll think about the psychological benefits of growing crops under extreme environments. And thankfully this movie came out. They didn't discuss with us at all about how to grow crops in space, and they did very, very well. The man at JPL who wrote the book and got Matt Damon to work in it, growing potatoes. The next question I asked to you: here is a spaceship or habitat, if you will, and they're growing potatoes. Would they have grown at all? Would there be, what would be the most limiting factor that he would have in this climate that you see right here? It would be like trying to grow potatoes in this room. We think there's a lot of light, but our receptors, our eyes, are significantly different than the receptors in photosynthesis, and you need the photosynthetically active radiation. The PAR in here is probably 5 micromoles, maybe 10 at most, because those lights are designed to do other things. And NASA, no one can afford a high amount of electrical lighting in any location. So there's, it would basically have been their working area, which would have lights even less intense that are here. Anyway, the potatoes would have never grown, and then the whole story would have gone down the tubes. But, okay, I thought it was great. It brought a lot of excitement back to food production and where we might go with it, where these young people might go with it. By the way, I was young once, and I thought that we would be on Mars by 2011, and that I was promised, and then it was 2018. Okay, I hope my great-granddaughter gets there. We'll work on that. Anyway, This is what we try to teach about technical success, about production success. We need experience and labor to be able to do it once we have the technology. And the technology is here today. It will be improved, but it exists today and it is operating. If you eat a fresh market tomato that doesn't come out of your garden, more than likely it came, 50-50 chance it came from a controlled environment greenhouse. Keep that in mind right now today. And when I first started in the late 70s, no one said we would never eat a tomato coming out of a greenhouse. Why in the world would we do that? Okay. Look at some of these annual yields. This number, the 435 tons of tomatoes per acre per year. That may resonate more than the 100 kilos per square meter, but that's the equivalent. And it's a lot of tomatoes. And then you can bring it to locations, in this case, urban agricultural situations. Again, to complement production agriculture in the field, not to replace it. But there it is in Brooklyn, overlooking Manhattan in New York City. And you'll come to find out this is Gotham Greens, and it was started by one of our women who graduated from our program and teamed up with 2 others and created this facility, and we'll look at numbers about that in a moment. The food, energy, water nexus and the interest that the USDA has right now is very important. Marketing, whoever was speaking about marketing before, really rang a bell to me to sell something. It's the buzzwords, it's the keywords, it's what your product can represent: inorganic, pesticide-free, locally grown, grown with fish, aquaponics, greenhouse. Anyway, along those lines, that helps sell the food. But if we look at where our lettuce greens come from, that's Yuma, Arizona in the winter. 90% of it comes out of Arizona. We also have, and others around the U.S., the greenhouse for hydroponically growing lettuce, not in the soil. And now there's these vertical farms using the pink lights, the blue and the red combination of LEDs that make it look pink and actually make the crop look pretty terrible. So add a little green LED and it looks an awful lot better. Anyway, the point is 90%, 10%, and less than 1%, almost 0, is coming out of the vertical farms, although that's the new and growing area, no pun intended. It's expanding outward in the heart of cities, and time will tell about that. But the greenhouse lettuce industry, that 10%, will continue to grow because of its capabilities of production and quality and consistency. Okay, here's where I make peace with everyone here. I have to say, and I say that I didn't make up this slide just for this presentation. I use it all the time. The fundamentals for plant growth: genetics. It's the genetic potential of the crop. Without that, we cannot do anything as an engineer or a controlled environment, no matter what environment we put there. But we can take that potential of those genetics and optimize it as best we can every day of the plant's life, as well as we understand it, and produce an excellent crop. The 2 environments are the aerial environment and the root zone environment. Don't forget about that which we cannot see. The parameters that you see here are the typical of what is provides the aerial environment. I'll make note that the last one listed is access for labor. That's certainly key to the aerial environment, and the fact that the plant needs to stand up somehow, and if it's not grown with its roots fastened to the earth or the soil or a container in some way, it will fall over in a hydroponic system, so you need physical support. As far as the root zones, maybe the less obvious, the temperature and dissolved oxygen. You can grow anything in water as long as, almost anything in water, as long as you have the dissolved oxygen available to keep the roots healthy and white. Water movement's a benefit. We'd like to keep it dark to keep the algae from growing, but I've grown them in direct light as well in aeroponic systems, for example. But here we are, one of my students, very strong, harvesting a whole table full of lettuce at one time. Components for hydroponics, and again, you're going to see a common theme here. To provide for the plant, we need this kind of hardware that will pump the nutrient water out to the root zone of the plant, have the contact with the root zone, and then recollect all the water that the plant doesn't use. Bring it back to storage so that it can be used later. And you better put some sensors and controls into the system so you have some real-time information about what's going on. But that's how, as in this picture here, you can grow a tomato crop without soil, without earth. Aquaponics, I put that in there. It's something of interest, utilizing fish and their waste in a closed system that you can produce the fish without having to discharge the water that they live and swim in, and essentially will kill themselves because of the amount of ammonia that they produce. Yet that ammonia can be converted to nitrate so that the, in this case, the Swiss chard could be produced. So it turns into a nice closed cycle, which then saves water in the long run and provides for, in a closed system, environmentally controlled, predicted facilities that produce the crops year-round. However, nothing is for free. You see that closed system, but we must not forget about the fact that somebody has to feed the fish, and the fish need protein. That's where the N is coming from to end up for the plants in the nitrate. So we need a protein source as fish feed, which is not always available, so something to think about. We need electrical energy to keep the system operable in pumping the water around, and there's a significant amount of that which it can never stop, in part because of the need for the dissolved oxygen in the water for the fish and for the plants. And of course we need sunlight in this case. No artificial light, we can use sunlight for growing the vegetable crops. And this happens to be a grower in the middle of Tucson, Arizona. He has a 1/4 acre facility that's producing these crops for the local school system, basically. Then you have to build a generic growing and climate control system around it. You can think about, like in the picture of this greenhouse, but you need some kind of a structure, some kind of a boundary around that aerial environment. You need light either from the sun or from electrical lamps. You have to heat. You will definitely be cooling in almost all circumstances as long as the sun shines and you have a well-insulated greenhouse. Humidity control, yes, for disease suppression. Also internal transport. The plants do not get there by themselves. They do not leave by themselves. That takes labor. The products from the plants might be continually harvested 2 or 3 times per week, such as with a tomato, a cucumber, not so much sweet pepper, but all of those then need access for people, and you need to make the labor less laborious, at least as efficient as possible. But you see that it's clean and it has safety concerns for those who are working in the system. In this case, it's indicating a top drip irrigation system, which is a dripper at each plant, providing X number of milliliters to every plant at the same time with the same nutrient formulation and draining away roughly the same from each. There's a uniformity around this monoculture, and it guarantees a consistency at harvest. The types of controlled environments, so you already looked at the high technology one, the very high producing Eurofresh on the left. Now it's owned by Nature's Sweet, same greenhouse producing tomatoes. There's the other extreme, the high tunnel. High tunnel greenhouse, hard to say it's a controlled environment because normally it has no heater. Normally it has no ventilation, no electrical ventilation. You roll up the sidewalls manually. So that's semi-controlled. It is a rain shelter and a windbreak, and maybe a provider of a little bit additional heat units early in the spring, and maybe some protection from frost in the fall, a little bit longer than if the crop was outdoors. The bottom line is it has a production capability and capacity and a price tag. That's reasonably matched. That's why it's being used. It's beneficial to the open field vegetable producer. Just as the high technology greenhouse has a super amount of productivity, but also a very high price tag, an order of magnitude greater than the high tunnel, at least, if not double that even. But that's the options you have, depending on the crop you wish to grow. In Japan, they call them plant factories. We try to avoid that term here in the United States. But essentially, it is because of these reasons, minimizing the use of land, maximizing the water use efficiency to provide fresh produce, pesticide-free. And in fact, I don't think I have the slide. There's a picture with Japanese writing on the wrapper of the lettuce. It says, please take home and eat and enjoy. Do not wash. There's a possibility of contaminating your salad by using the local water. You have to keep that in mind. So there's times and place, and at multiple levels using electrical lighting. In the U.S., it's been called vertical farm, which basically is a series of shells growing in the vertical direction with electrical lightings at each level. Very high potential yields. Business viability remains unclear. It is to be established, but it has the potential. Primarily what happened, these lights called LEDs came to be much less expensive than they had been in the 1980s and 90s, and in the early 2000s just took off and now will continue. The benefit is that they can continue to become more efficient in the future, where fluorescents and high pressure sodiums that we're using now have pretty much reached their peak. So I expect the weak point is still the cost of electrical lighting in these systems, but that's the challenge we have to try to improve and reduce those costs. And I believe it will come, and it'll be used in locations where it makes a lot of sense. Not necessarily where you have the Yuma lettuce fields in the backyard, but maybe in the areas of Alaska, remote locations where fresh produce is expensive and it's cold out there. Although the light shines a lot in the summer, it's a long winter. But even locations like Hawaii, isolated from the rest of the world by the ocean and having expensive resources, this allows you to use them more efficiently in the long run. Anyway, we have a unit that we're studying, Dr. Kachira, my colleague, and others, the students working on this at the Controlled Environment Agriculture Center. Here's Gotham Greens again with Jen Nelkin, and she is the grower, essentially. It's a nutrient-filled technique, hydroponics. Each individual row is fed by a dripper and the water flows down and the plants are sitting in this little trough with a very shallow film of water over there. 45, in her case, 45, up to 45 lettuce heads per square foot per year. And there's been other systems that have reached 55 and 60 heads per lettuce per square foot per year. It also depends how big of a head of lettuce you wish to grow. We're not talking about these massive romaine heads that you got to carry like this. Smaller, 100 grams, 120 grams, maybe 150. But in locations now, actually in Chicago, started out in New York City, she's building another one in Baltimore. And in their greens, they can get an equivalent of $16 per pound. So it's a pretty high-end market. And here's one of their greenhouse facilities on top of a Whole Foods market in New York City, where the first time that the building underneath was built originally from scratch, from the beginning in design, to have the greenhouse overhead. In most other cases, it's fine a building that has a big flat roof and not a lot of junk in the way, HVAC systems and plumbing and all that. But there you go, and they certainly market right below, as well as to other places around the city. And at this point, still never seem to reach their, to satiate their market demand. That may come, but not at the moment. Let's look at an application at the South Pole. This is a project that we have with the National Science Foundation, who, through our tax dollars, runs all the research that goes on, or significant operations of the research that go on at the South Pole in Antarctica. And we won the contract to design, build, and then help operate this food growth chamber. It's about 240 square feet. It's a closed system, hydroponics, recirculating water, because water is somewhat dear at the South Pole. It's all there as ice and snow, which takes energy to melt. And at $35 per gallon of diesel fuel, you don't want to be burning too much to try to melt water. Anyway, we had to be efficient with water use, recycle, use the least amount of light possible, and have this system produce fresh vegetables, not for research. This was for production at the South Pole. Can you see the precursor of why we then later could move into the Mars Lunar Greenhouse? Extreme condition, limited resources, not a lot of connection to the outer world. This place keeps operating all winter long, their winter, our summer, with 60 or 70 people that are trapped there. They cannot leave. The planes, it's dangerous for them to fly in, and only in great emergencies will they, and certainly not in the middle of the winter. So to have fresh vegetables through the winter since 2004, this has been operating, and it continues to operate today. One of our students, actually, Lane Patterson, I say he went down there and made it work. We put it together and tested it, and he was part of the design and testing. But he lived there a number of times, actually, and was able to produce a significant amount of product. Everybody got a salad every night, which was impossible in years before. To take a look at that from above, it's 3 rooms. The plant growth room itself, where we provide the environment that the plant needs. The enviro room, and can you imagine what the enviro room is? That's the place where people who've been living in the dark and smelling only diesel fuel and only showering once a week go to get away from the fluorescent lighting of the building and see high pressure sodium lights that look like the sun. The place smells better, and it's warm and humid, which Antarctica is a frozen desert. Less relative humidity than we have in Tucson, Arizona. They're down in the single digits, the low single digits, so a lot of nosebleeds. Life is difficult. It's an extreme place. And then the utility room, where we have to have our hardware systems to keep all the plants alive. So with the production room, we see that there were the tall plants in the middle row, and then there were the smaller plants, statured plants on the sides, with a tray that moved in and out. So we used every square foot of walk area in there for plants. When you wanted to walk in, you actually had to move a row of plants to the side. And then there were 2 lines of 6 high pressure sodium lamps, 1,000 watt high pressure sodium lamps above on either side of the tall crops, efficiently lighting both sides of the room. And then we used telepresence. I never went there. They said to go there you had to be useful. I guess, you know, oh, we just designed and built it. But you send the students because they are useful. They can get experience, and they love it. And I went to Tucson for a reason, and it wasn't to be cold. So through telepresence, here's the view. You don't see many food service opportunities beyond this structure that you see here. This is the South Pole, and then there's the picture of the food growth chamber, which is located inside the building of the South Pole. So we're not at the whims of the tremendous weather there. It's in a 10 degrees Celsius building, but we still had to grow the plants. So what do we do is that there's Lane, and he was already pretty hairless when he got there. That's not the reason why he's like that. He's down there working, and then there's another student, and then Phil Sadler, who actually built all this. Small businessman from Tempe, Arizona, craftsman extraordinaire, built the whole facility. We just made it work. That's what I say. He let us turn it on and run it and operate it and put the nutrition into it and the lights and all that. But he made the HVAC system. But every week we'd have a discussion with the South Pole to try to convince the people that, yes, we were monitoring and we know what the environment was last week, and we had a web camera there and we saw what the plants look like. And yes, we can help you with some advice on how to proceed forward. Until they had access to the internet, when they said, well, on the internet it says. So you can't always compete with the internet until you allow them to try it, and then maybe they come around and learn. It's all a learning process. This picture told me that we were received by the people living and working there. To a married couple that were celebrating their wedding anniversary with a dinner special in front of the food growth chamber in that environmental room. I guess they kicked everybody else out for the night. Environmental room is only 14 feet by 7 foot, so it's not very big. And we take no credit for the glass of wine that was there. We're not growing grape down there as yet, but maybe someday. And then you get this. That is the true South Pole. And that's Lane doing his funny thing. But that is the pole at the South Pole. If you get an opportunity, maybe you can see it sometime. All right, the Mars Lunar Greenhouse. I'm finally getting to what you asked for in the first place. Why? Well, it's another challenge. We can grow anything, anywhere, anytime. Okay, let's talk about the Moon and Mars. Did we do it? No, we're doing it in a laboratory on Earth, so we're not exactly there yet, of course. But you should be able to see some of the similarities of a system that would go. The justification that I could provide to the dean when I get questioned on occasion, why space, why Mars, why not Yuma, what's going on in Arizona here or in North America. And I can safely say that this is just growing another plant in a controlled environment that's using technology from the late 1990s that went into this. And we're going to learn more from it to bring back technology and recycle it back into growing systems that we have here on Earth today, feeding people, and give them some of the numbers along those lines. And under stressed resource conditions, particularly water, particularly lack of labor availability. So automation is important on the other planets. The lack of pressure, oxygen, obviously. But bottom line is, it's collapsible, it's lightweight, so that it can be shipped relatively easily. And it could provide vitamin and minerals. These are vegetable crops. I'm still thinking, at least in our scenario, that even at Mars distance, to ship the flowers and the processed grains that are very energy intensive to grow, like the corn and wheat and rice, but grow the veggies that provide the fiber, the vitamins and the minerals that are difficult to obtain in a flower, for that matter, and then recycle everything. So that was our goal. This is, I assume this will be available online at some point, maybe, but that link is something that you can go to, and you can hear me telling another story specific to a video that we created. But here's Phil Sadler's. That's a 1,000 watt high pressure sodium lamp, water cooled. So you could touch it. You wouldn't want to keep your hand there long, but you could touch it, and you would not be damaged, burned in any way, which also meant that we could grow in only a 3-meter tall location plants right up to light, as opposed to having the non-water cooled, where we could at best be able to just do a horizontal platform on the floor. So this was developed by Phil prior to going to South Pole or going to the NASA project here. And he was looking for a prototype that was relatively robust, or as he says, high fidelity in terms of its representation. It's NASA-esque if you look at the geometry. And like I said before, it's lightweight and it's collapsible, so it could be shipped. And in theory, with the automation that's available today, think about the Mars rover, think about the spacecraft that's going to the asteroid to bring back a sample. We could automate the process of delivering this to the planet in advance of the astronauts. It could be deployed. It could potentially be operated so that when they arrive, there's actually a crop there, and that's part of their life support system. Not all of it, but part of it, because there's always backup systems. So that was the concept. Here's the dimensions. You didn't see any people inside yet, but it's about 18 feet long, about 5.5 meter cylinder, 2 meters in diameter, about just under 8 feet. So you can stand up when you walk in there, and you don't singe your hair on the, at least I don't, on the hot lamps. I do have students that, yes, that they had the challenge. Third reason, provide food. Second reason, provide water, or water that can be recycled as nutrient hydroponic water that the plants could transpire. And then third, to provide food, but also fourth, psychological benefits, to be able to live and work and see a green plant where you're living in a spacecraft or a habitat on another planet. Here we are when the plants have been established, and here we are about 40 days later, and we like a lot of biomass. Particularly, we're trying to get as much oxygen generated as possible. And obviously part of our challenge was, at least in this one, was not to try to determine how to dynamically time the growing of the crop so you could have harvest for food, but not lose your life support system if you had a big meal the night before. That could be a challenge. We were just looking at, and I hope it shows here. Okay. We were just looking at the fact that we would input labor, carbon dioxide, water, nutrients, and electrical power for the lights. And out would come food, oxygen, and fresh water. And we were doing that balance and saying, does it work? Is this big enough for one astronaut? And that was the expectation. And the answer is no. It's only about 10 square meters. Ultimately, the minimum is probably about 20 square meters per astronaut. And if I was going, I would want it even a little bit more, for sure, just in case. So a couple of things that were designed were the water-cooled lamps, shown here, and the what's called cable culture recirculating hydroponic system. This is the nutrient film technique that's supposed to be horizontal, turned vertical, so it could be hung on a cable. And why do we want to do that? Because it's the lightest weight. The material is made out of thin film plastic, lightest weight trough possible. It's a collapsible trough. It could potentially be seeded in advance. Yeah, it's gone a little too far, but okay. And then it could be expanded out and take off. So there was this preliminary work done, and there we are. You can see Lane's face in that way up there. So that's a typical door hatch, and you see lettuces, you see cowpeas, you see sweet potato. In this one, we have grown strawberries certainly, and basil in some of ours crops that we've grown. Lightweight collapsible hydroponic system, something that could not be done in a soil-based system. And by the way, there's no soil on the other planet, on the other planets. There is material that could be similar to soil, but would you trust your first crop with something you can't even do the chemistry on in advance? So that will happen. I'm sure it'll become a soil system at some point. Keep in mind that even in a hydroponic system, we cannot eat all the biomass. There is inedible biomass, and eventually you create soil. We're going to make compost from inedible biomass and ultimately recycle nutrients as much as possible. Anyway, it makes some sense, but ultimately that it's going to go to from hydroponics into a soil-based system, but we can still keep doing hydroponics along the way. So there's a heavy crop of lettuce on that. Why now? Why now? The word national security has come up, investing in science and securing our future relative to food. I don't remember us being too concerned when the rest of the world had problems in the 1980s and 90s, significantly, about where all our food was going to come from to feed all these people. But that seemed to have changed after 2000. So food is exciting in this century. I don't know. Is it the youth? Is it you millennials out there? I mean, my youngest son is about at that age, and I question him now and then, and you know, I don't get an answer. So, but you know, they want easy access. And how about for us old folks? I want good food. As long as I'm around, I want to be healthy, and I want to enjoy it 3 times a day. So somebody's going to have to figure out how to grow food, because I want to eat. That's being very, very selfish. And then the tech people have come around. They need a place to put their money. Let's get into agriculture. I'm sure there's a good rate of return on that. No laughs in the audience. Okay. Anyway, yeah, well, they get warned, but when you start putting $200 million into a company called Plenty and to grow indoors, you can go a long way up that learning curve with that kind of money very, very quickly. We'll see how the returns come back. I have all expectations that, just as in any businesses, some fail, some succeed. But it's all a learning process along the way, and that's what we need to do, and keep supporting them. So we do, as much as possible. Just some examples. You can look around and find monies that have been put available just in the past 2 years, and it's constantly increasing. And then our former director of NIFA spoke about CEA. I put it in red over there. That's his slide. Finally, USDA, recognized and controlled environments. Initially with high tunnels, but okay, controlled environment, agriculture as well as part of the toolkit. You can walk through our teaching greenhouse some other time. Why CEA? I think you may recognize these, that of what I've been talking about in terms of efficiencies, but look at location, functional on poor lands, complementing field production, captures the imagination of young people, those who have never grown a crop before or had the opportunity to. I say now, this is the first generation of people ever that came from a non-farming background that can become a farmer and be profitable at it, not just feeding themselves, but feeding others as part of a business, because of hydroponics and because of controlled environment. I think that's just great. Sociological changes, just a couple here. Year-round jobs, professional, certification, higher salaries, benefits, communities based around 12-month per year agriculture, as opposed to seasonal agriculture. Working conditions, locally grown food. All these things can benefit our society. And here can benefit our environment. Zero, I love these zeros. Zero erosion, zero nutrient runoff, zero pesticide, maybe. You can do it pesticide free. Not always. Integrated pest management works to a great extent. It's one of the tools in the toolbox. Zero herbicide, yeah, okay. Sorry, weed people, there's no weeds in our greenhouses. If there is, we're doing something wrong. So you're welcome to visit. Just make sure you shower and change before you come. No soil-borne diseases. Anyway, therefore, we're getting near the end here. With knowledge, technology, the flow of information, that means education, with resources, marketing, and a food chain that can deliver this product efficiently, and with these investments, we believe we can help feed the world in the future, now and in the future. Certainly we can provide more nourishment for the world than exists at this point in time. I leave some summary of challenges. Education, not just of the producer, of the young people or those who've been in production or want to get into it, but the consumer has to understand what they're buying and why they're buying it. as well as a distributor to know how to deal with it to keep the quality. Water and energy is obvious that we need to continue utilizing what limited water we have at the quality level that we have it, or improve it somehow along the way. Energy sources, yes, we need energy that's not just solar for light, for photons to grow plants, but electrical power from solar to help grow plants. Nutrients, the question of organic versus inorganic. You can tell me, can the plant tell the difference between a nitrate molecule that came from the bag in the storehouse or came from the compost pile down the street? I'd love to hear that discussion. Finances and market and social development, yes, they will power change, and the social development will make that change stick. Last slide. We went to the moon. Some of you may not realize that. People walked on the moon. And I love the way this was presented. I didn't create it exactly, but I found information. Some president made a commitment that 8 years later, this NASA group would land someone on the moon. Then the next president committed 5% of the annual U.S. budget to this project. Can you even imagine 1/10 of that in these days? But 5%. So that a technology that didn't even exist, many of the technologies, including the rocketry, let alone the life support for the people, didn't even exist. And it was all done with NASA engineers and businesses around, well, I speak to NASA, but obviously the businesses around the country. Young people having the average age of 24 years. Think about that. I mean, that is significant impact. There's a whole generation of people that have to be very, very proud. So I throw this on the side there about youth being a state of mind, and pardon the double negative, but not knowing what you cannot do. Let's try it. And that's why I say we will continue, and hopefully all of us will eat better in the long run, and the environment will improve as a result of that as well, and that our society will continue to flourish. So with that, I'm going to say thank you very much, and I would appreciate if there's an opportunity to have questions. Otherwise, thank you very much. Yeah, I put a lot of web references to our work and other people's work. So if you can get to that file, you can find that information.
Exciting. Can I hear, I've heard from students that want to use recycled container shipping, and how efficient is that? Is that something doable, or?
We use recycled containers?
No, the shipping containers.
Oh, shipping containers, yes. Well, there are companies out there in the real world that are using the shipping containers as production facilities. And they work. It's a controlled environment. You can make it happen. It's maybe not the most uniformly controlled environment, but I have friends in the business that would say, come on, Gene, you know, we can do it. And they can. The question I have is that amount of productivity in that unit sufficient for your application? And I think it can be, like if it's at a school and they want to teach, or at a hospital if you want to have some fresh veggies. But for a commercial production facility, it's too small. And by too small, I mean there's too many edges and corners and doors you have to walk through to see and to work. You can't have 10 acres of them. It just wouldn't make sense. Unless it was fully automated, roboticized, but if you could do that, then why not just do that in a greenhouse or in those large vertical farm buildings? So the containers are nice because they exist, and they can be adapted, and they will work. But they're not perfect for all applications. Are the students building one here and we're going to make it work? Oh, you don't know. A group did at Arizona, and we have one that's available for people to see and walk in, and they grow crops in it quite often. So to me, it's another controlled environment. I can find all the reasons why it works, and I can find the reasons why it has some concerns. Like with any growing system, it comes with the territory. Did I answer your question? Maybe too much.
My question was a little bit more general. I was wondering if breeding for the expected flavor of these crops is still a major issue with hydroponics, or it's kind of moved on to bigger, more efficient.
No, I don't think it's moved on. I don't think it ever arrived. We need breeding for controlled environment crops. Right now, there's people out here that know more about it than I do, but my experience with crop breeding is that unless you can sell tens to hundreds of millions of pounds of seed per year, it's not worth a company to invest in doing it. So you go after the big ticket items, and we know what they are, which feed the world. So I'm not arguing that point. But there are smaller companies, particularly in the Netherlands and Holland, that you can buy seed with a Dutch name. Enzazeda, for example, is a brand name, and there's others. And they bred lettuce and tomatoes, for example, specifically for controlled environments. And if you think about it, there's different environmental concerns that controlled environments plants need in their genetics, and you can focus more on the quality of the tomato or the head of a lettuce or the phytochemical composition. The anthocyanin is something that you're interested with, the amaranth, right? Same with the purple or the dark, the reddish lettuces. Same thing, that are being bred just for that. There's a researcher at Rutgers University that released one lettuce 2 years ago. But that is important, is needed. Right now, we take what is available and work with it. Plug it in, turn it on, see how it responds, and hope for the best. But the first thing that is done, and I'm ready for the next question, is grow it and find the quality of the harvestable product, because that makes your product a premium, and then it can compete even with the summertime grandma's garden in the backyard, because it's a brand, it's consistent. If you like it today, you can like it next year, because it'll be grown exactly the same way. If you don't like it today, then we're in trouble.
I'm
Yes?
wondering if you are working on greenhouses that can be used in regions that will definitely need to be fed. With that, I mean developing countries. So are you doing anything in your research program? Because most of the tech that can be used in the developed world obviously won't work in developing countries where we have not access to electricity or just the equipment and everything else. Thank you.
Good, yes, very good question. And that's a challenge because of local resources. So you use local as much as you can, but if you want a controlled environment, then you need a glass cover. That's tough. Or a plastic polyethylene film cover, maybe better, lower cost. And we have had projects that are sustainable for locations where there is no electrical grid. And what is needed is a place that has sunlight, that has potable water, and animals around so that you can get some fertilizer. So yes, absolutely. And they are closer to the high tunnel design that I spoke of, lower cost, can be more challenged under difficult weather conditions, but offer some improvement over the open field at that site. And that's been demonstrated, yes. By the way, I just have to mention something, and I want somebody to take my picture in front of it. The name of this room is Monsanto. I love it. And the reason why I love it is, well, you also, about this project, that they were Monsanto, and I guess we did so well, we put them out of business. I don't know what happened. They became Bayer. No, I can't take any credit for that, for sure. I'm not a businessman. But in my early years, I came to know a plastic film, and I thank you for asking that question, that was created and was called Monsanto 602. And I don't know the numbering scheme for sure, but the 2 seem to represent that it was the first polyethylene film plastic that would last more than 1 year. And in fact, it would go through 2 growing seasons. And it took the high tunnel industry that existed at that time, that did have heaters and some fans, and allowed growers to not have plastic that would fail after 6 months. So Monsanto was in the plastic film business. Now that went away, moved on to other things. But that, to me, was a jump start in the that was early 1960s. Just a little before my time, but I had mentors that could tell me that story. And I can tell stories too. So I see that as critical. So I'd like that picture. Nothing like a guest coming and making demands, right? But I've enjoyed the opportunity to be here. It's a privilege and it's an honor to be here, particularly all the people that I heard speak today. You have passion about your work. I congratulate you on that, and please don't stop. Just keep going. Thank you.
So we've come to the final presenter of the day. One of the benefits of having a sponsor like Corteva is that they provide high quality speakers for these events. So it's my pleasure to introduce Dr. Rodrigo Soria. He's the input trait discovery leader in trait discovery at Corteva, so that involves trait discovery and implementation around insect control and herbicide tolerance. Please welcome Dr. Soria.
Well, thank you very much to the organizers for the invitation. Not only for that, but I also congratulate them on, as the previous speaker said, it's a great panel of speakers today, and so got to learn a lot about different things that I normally don't come across in the job that I do. The topic of my presentation today is going to be the request was how the discovery pipelines in industry work. How are they structured? How are they organized? And so what I thought was that given that we are a relatively newly formed company coming from heritage organizations with a long history, I think we are currently positioned to do some things better than what we normally have done it in the past in our respective heritage organizations, and try to create a new perspective. So what you're going to be seeing today is going to be framed in the context of what our new organization looks like, what that structure is. So I'm going to take you from what you probably will say is a corporate speech, but I consider that very important because you got to anchor your passion and what you do to where you are and the mission of the organization you work for. So I'm going to take you on a journey from that high-level corporate perspective to then explain a little bit how we contextualize our research and discovery platform in R&D. And then I'm going to give you some examples of the types of things that my organization, my group does in terms of input traits and insect control and herbicide tolerance traits that we work with. So as I said, it's important to contextualize, and Jesse this morning referred to our purpose statement. What our purpose is, I find it very inspirational and enriching lives to those, enriching the lives of those who produce and those who consume, ensuring progress for generations to come. And so I think that kind of explains a little bit the context, the societal context that our organization tries to have, not only as a commercial organization that it is, and that is a for-profit organization, but also on a mission that we intend to have with consumers, with producers, and with society in general. That's important. That's important to anchor to that. And with that comes some values. Those values are enriching lives, stand tall, be curious, build together, and be upstanding. And so those values refer to multiple things. So I find them refer to multiple things like innovation, the role in society, developing our people, working in teams, being an entity that integrates in general to society, and to which we do good not only for our employees but the public in general. So how is that, with all the challenges that we have currently in agriculture, we are positioning ourselves to lead the way for the future of agriculture? And so being an industrial organization, it's important that we always keep in mind things like value chain, that we anchor, these processes that we have in order to deploy a product in the market. But probably even more important is what you see in the middle section, that is related to providing innovative solutions. And that's where I root my beliefs more deeply. That's what my groups do. That's how we find motivation to do what we do. And more importantly is that once you develop these products, once you have produced value, you also have to be responsible with that product that you have generated and steward it properly, and make sure that it's safe and that has no issues. Talking about that role that we have, that is to increase productivity and food outputs in general. If we think a little bit about how agriculture grew in the past, was by increasing the number of acres that we had. But now, over the last century, that has started to change. And so now the way in which we increase productivity is we don't have any more arable land to colonize or to generate. In fact, we have to think more carefully of protecting already the already scarce resources that we have in terms of biodiversity and protected spaces. And that's to avoid the imbalance in the planet. So what is the challenge for us today is that we need to produce more outputs per every area unit that we have. So that's the challenge, is that amount of land is not going to increase. It's probably going to be reduced if management practices don't erode it and erosion doesn't become a problem. But the reality is we need to increase output per area of land. So, and there are together associated with that, there are some factors that are limiting to food production, and those factors are listed there. Those are all the negative factors that go against food production in general. It's a very long list, as you can see, all the way from biotic, abiotic stresses, management practices, agronomic practices in general. And but the ones that I'm going to be that are going to be the top ones, and some of the ones that impose more pressure on food productivity or crop productivity, are those three. And my group is primarily associated with the top two, so that's what you are going to be hearing more about today as we progress in the presentation, is the pressure of weeds and the pressure of insects. So in order to tackle all these complex problems, and the way we have gone about is to take a very comprehensive approach with multiple technological platforms. And so you have heard today about some of them, but are all the different versions of breeding that you can think of, all the way from the targeted breeding to in silico breeding that nowadays is possible, to the regular and conventional breeding, biotech tools like what I'm going to be referring to today, crop protection tools that are code word for agrochemicals and chemical solutions, seed applied technologies, digital solutions, and all the agronomic solutions. So those are technological platforms that we have in our organization, and when we are looking at a problem, we try to look at that in as many ways as possible. What is the most convenient? What is the easiest and cheapest solution, the more beneficial solution that we can deploy to the farmer? Then the second thing is that it is important that all these solutions work in concert. So the problem unifies the use of the solutions, but those that are responsible for the technological elements of how we use those solutions have to work in concert. And that requires not only that they work in concert, that they work in teams and in very unified ways, but also that pipeline of solutions is robust, because we know in R&D that the number of winners that we get out of everything that we tackle is very small. It's a very reduced number. 90%, 95% of those things fail, and a very small percentage really gets to succeed, and we get to provide as a solution in the marketplace. So here I try to illustrate not the whole pipeline that we have, but what has been released in terms of products in the last few years, and probably starting 2016 or so. And they are divided in 3 clusters, and this is what the 3 clusters of the unified Corteva organization look like. And so we are bringing seed and trait solutions, we are bringing in crop protection and digital solutions with our 3, let's say, technological platforms in our research and development organization. The recent launches that you see there, I'm going to start focusing more and more on the seeds and traits, because that's what I'm going to be taking you to. So that seeds and traits organization has focused primarily on insect control solutions and herbicide tolerance solutions, because, as I said in one of the previous slides, that has been a major focus, and solutions for those were among the first ones that appeared in the marketplace, and that carried on until very recent years. As years progress, you see that there are more and more diversified solutions in other areas outside pest control in general. And so you see that in 2018, there are solutions like the unified corn silage. Those are native traits that have been identified, and they are optimal for cattle feeding, and especially in dairy, but also in beef production as well. Those are technologies that have been identified and developed using biotechnologies, but not transgenics. And so that's where all the genome sequence and all the molecular marker information has been deployed to develop these type of traits. And you see the Intacta traits that are insect control solutions that we have for soybean. Plenish, which is a type of quality trait, and Enlist, which is our new herbicidal or herbicide tolerant solution that we are deploying. And it continues to go on into 2019 and 2020, the 2019 and 2021 time frame, as those solutions continue to get diversified more into, we maintain our focus on pest control, but we also maintain an emphasis on quality traits as well, but now expand into multiple crops like canola, going beyond corn and getting into soybeans, canola, and sunflowers. Then going forward, we intend to maintain that diversification, and that is the type of pipeline that I'm going to be giving you some examples today on the sorts of things that we are trying to, the challenges that we have in doing that, and also the type of solutions that we are thinking of bringing to the farmer. So this is now more focused on the insect control and herbicidal types of traits. This is exclusively for that. So these pipelines have evolved in multiple ways. So it started with what you see there as a first generation where there were single mode of actions. The solutions that were offered were single mode of actions. There was one mode of action trying to target as many pests as possible, trying to have broad range, and obviously being limited in that reach. And as the second generation that started circa 2010, what you start seeing in the marketplace, not only in our company but in the marketplace, is more of the pyramiding of the traits, and so stacking up multiple modes of actions to try to have a more reach in terms of the target pests that we were trying to address. And not only that, but also on top of all of it, we were having big ambitions with maintaining at the highest level of quality in our germplasm, because some of these traits in the past, not only in our company but in the industry in general, have had some yield penalties, and nothing comes for free when you are modifying a crop. Even though it's a highly precise technology, there are also minuses that come with a technology. So it's a compromise that has to be reached. And so in that second generation of traits, there is by far more emphasis in trying to make sure that crops don't suffer from that, and they come with as much, the trait is as potent as possible, but also the productivity is not affected from any, under no pressure, is not affected in any way. And then in what we are targeting in that third generation of traits is first, there are some positions, some traits that have been well positioned, but we are not naive, and we know some of those tolerances are going to be broken at some point. And we are not only emphasizing on our stewardship programs and how those crops have to be managed. There's a lot of talk that was this morning about integrated pest management in general in the case of herbicide, of weeds and herbicide tolerance, but it's the same case for insect control. And so we are thinking of what are the type of solutions that are going to be replacing those technologies that we have in the marketplace today. And then the third thing is that markets have become more industrialized outside the US. More and more in South America, you see that there is more demand for these type of traits. Agriculture has reached new levels. Farmers are becoming more knowledgeable in those geographies as well. And so there is a very high demand for quality. The caveat to that is that some of the pest pressures are also different. And so then a single product is not going to work in all the geographies, in all the environments. So going to be a customization of that product line for different geographies. And so since that, the request was, how is that the discovery pipelines work in industry? And I, throughout the day in different conversations, there are always these questions about why did you decide to go to industry? What is it to work in industry? And how is that things get planned, and you know, what the culture and environment looks like. So from this moment on, I'm going to start kind of illustrating how is that we see things, how is that we can also, I can give you a little bit of a glimpse of how you can do that, some of that work yourself, and how you can go and figure out what industry is doing and how to assess what industry is doing, the different companies and so on. So this is an example that comes from the reports that we go ahead and do to the investment community every quarter. There are reports, and with those reports, if you go through them, there will be a portion of those reports that are going to be reporting things like what you see here. And what you see here is a slide that was taken that outlines all the different product concepts that we have outlined for corn in particular. And so what you see there is that there is a list of product concepts that we have. Then in the next column, you see what the R&D phase is, and a bunch of numbers 0 to 6 that are listed there. And I'm going to go through all that. And then you will have what are the markets in which that every specific product concept goes to, and also what that market opportunity looks like. And that's because investors like to see what the benefit of that technology could be for the organization. And then that last column is the estimated peak sales. So that's whether that's at peak sales you are going to have high, low, or medium profit from that. So you see that when we list a crop, we also list the type of product concept that is. So that is whether it is a biotech crop or is a digital solution or what that is. So I'm going to go ahead and start talking a little bit about what those 0 to 6 stages look like. And so what you see here is the staging process that in general industry follows, and I'll go one by one and explain what that is. But let me start at the top, and so there are two distinct phases or big stages, and those are called discovery and commercial phase. So all the stages that we have between the discovery stage 0, and those are the numbers that you saw listed there, are 0 to 2 are what we call the discovery phases, and you see that the year duration there is in ranges because it depends on how complex the trait is. And when you are also stacking up traits, then you have a compounding effect in some cases. In other cases, you don't have it, but it's not so linear and straightforward. And basically what that is stage 0, by the way, my groups focus primarily on that 0 to 2 stages. And so that is where we stay in stage 0. We are looking for genes. We are looking for all the potential sources where those new traits may be. And when we say genes, we are now constantly looking for different modes of actions because we want us as a diverse mode of action pool as possible so that we can circumvent resistances in the case of pest control. Then when you move into that, and there are some criteria that you have to meet at the end of every stage, and this is vetted by a team of scientists, and in some cases scientists and people that is expert in project management, and also the business side of the organization. They are the ones that you see these processes there listed in a very linear way, but in fact there is a feedback loop that happens at some point because we are sensing all the time what is happening with the farmer, what are the needed solutions by the farmer, and all that information retrofits into our discovery teams early on. Then going back to the discovery stage 0, once we meet the criteria for promotion, then things move to what we call proof of concept, and that's when by that time we have already looked in the crop and tested or assessed if the phenotype, the desired phenotype in terms of tolerance or resistance has been met and identified, and that happens normally in the crop. Not extensively, but at least we have an indication that this thing, that this new trait has a promise to work in the crop. Then once those criteria and that characterization has been done, then that is when we move into using plant transformation technologies, and then we go ahead and start working on the development of what we call a commercial grade event. And so those are events for which when we go and analyze, we do the full-blown characterization, starting in control environments, starting in the lab, control environments, and in the field. So all those criteria need to be met, and so every one of those steps needs to go through a thorough characterization at that point. And then, when things get promoted to that, from that stage and they move into commercial phases, that is when, the commercial events get selected. Those events get introgressed into germplasm, and you see how the different arrows there start sprouting. Those are outputs because we go from having multiple candidates for a trait to have very single one at the end of that stage 2, and then from that moment what happens is that we are deploying that trait in as many lines or varieties on our target crop as possible. And so those are the green arrows. That's when we are starting our process of introgressing that new trait and testing it as broadly as possible and making sure that it works in as many lines and geographies as possible. And then all what happens afterwards is basically going into initiating submissions for the regulation. There is a very thorough process for the regulating these traits and assessments on safety and all sorts of different things that have to be done for every country where we are going to be deploying the trait. So it's a very, you want to call it tedious process in terms of making sure that we meet the criteria of every single country for every trait that we have. And then by stage 4, basically what we are doing is that first pre-launch, and we are preparing, basically bulking up the seed and making sure that we are going to get ready for the actual launch of the trait. And by that time, the regulatory approvals must have come up. Otherwise, it doesn't make any sense to, create large piles of the commercial seed as we are supposed to have. And then the launch really happens in a single year, and that's when we launch sales. And so when you hear the news, a new trait has been launched and all that, that's normally in the target geography, only one geography. That's what we do. Sometimes we do multiple geographies if it is, let's say, clear that the trait is going to succeed in all of them, or if we have the approvals in all those geographies. And then finally, the ramp up is really when you go full-blown commercial, and then you are having the trait deployed in all the different geographies, and that's what we call starting to have the market penetration, starting to go aggressively with the different markets that we are supposed to have. So when you go from, I'm now going to switch a little bit on what this means in terms of technologies, what happens, what is being used at each one of these stages. So when we are talking about discovery, really we are talking about going into gene sources, using a lot of bioinformatic tools to identify, to see from all that sea of genes that are out there in the different collections, in the different sources that we have, organisms, selecting all the potential targets. And we use not only genome sequencing, but we also use proteomics, and we just simply also go through data that has been generated, data that is in banks, data that is coming from organisms that are living under special conditions or biomes or different things. And then obviously if you have identified too many of those, then you will have to go ahead and test them, and for that you need to put them in constructs and be able to test each one of those genes. We resource to a lot of high throughput building of constructs and that sort of thing, and so lab automation tools have in addition to be able to build them and test them, then have the right ways to test it. And for that, when you are testing thousands and thousands per every target that you have, then you have to have also high throughput bioassay technology, the assays that have been developed for all the different pests that we have, and that we put all our genes through each one of those bioassays so we don't miss anything. Then when we have the proof of concept stage, at that point, what we are going to be focused on is in making sure that we are capable of doing characterizations in planta. This is not only in vitro. The previous stage can happen in bioassays. Those are Petri dishes or small formats, let's say, that still happen in labs. But for proof of concept, we already need to go for full planta, full plants, or whether that is in models or directly in the crop. It depends on the type of assay or stage at which we are. And obviously, we resource a lot on lab automation. There's a lot of characterization that happens of these materials also at this point. We need to keep track and check constantly that things are as they are supposed to be, especially when we find potential winners in these pipelines. And then resourcing to phenotyping technologies, that's something that has become more and more important, and is not only from the perspective of the trade, but one of the things that we try to make sure is that there is no agronomic penalty, that the architecture of the plants, that those, especially in the crop, that those plants don't look any different from what they are supposed to look like. And we do that in control environments, and we do that in the field. And so hopefully what we try to do all the time is that the type of characterization that we do under control environments is as rigorous as possible, so that we try as closely as possible that we are evaluating the things that really matter in the field. So we are able to identify the characteristics that are going to be predictive of this is going to do like that in the field, sort of. Then in the early development, as I mentioned, so it's all about really the commercial event production. So that is the construct that is going to be put in the plant, the configurations that we are going to be used, and the initiation of the regulatory studies that we need to start providing. And so high efficiency, high throughput transformation capabilities are a must at this point, because even though the numbers by this stage have reduced considerably, the number of leads at these stages are still large. And that is because we kill only things that are obviously wrong in the crop, but not everything is black and white. And so that's the problem, is that there is a lot of gray in these type of things that we do. And so we need to learn and do a very good discrimination of what that gray means. And so large transformation capabilities are important. We do that in high throughput. Enabling technologies for gene expression are a must, is something that we constantly look for. We have realized how important is becoming to have the ability to have precise expression of genes, and that is something that we have just discovery programs around regulatory elements to make sure that we have a good toolbox to express these traits. And then there is something that you don't hear much about, which is gene stacking design and technologies associated with it. Not all the gene configurations in a stack work the same. Not all the combinations of a promoter, a terminator, and a gene work the same. And we do pay a lot of attention to every, let's say, configuration of 2 or 3 gene stacks. We try to test it in as many combinations as possible under as many as different conditions as possible to make sure that we don't miss anything when we put those materials in the field, to make sure that there is not going to be unintended consequences of these things. So we have done on occasion combinations of 3 genes where we easily can put together 300 vector configurations, testing different things to make sure that things are as clean as possible in terms of expression, in terms of agronomic performance as well. Phenotyping technologies, obviously now at this stage is not only the emphasis at this point is tilting more towards the field. So we continue to do that in the greenhouses. We continue to characterize with phenotyping technologies in control environments, but we are more interested at this point in testing under field conditions. So that is a big emphasis that we put into. And now, after you have taken the opportunity to see what this staging looks like in an R&D organization, so what is going to happen, and just to give you kind of a general idea, because I'm not going to cover the commercial phase of things, what is going to start happening from that moment in which we select that commercial event is that basically the testing across multiple commercial varieties is going to start happening, introgression is going to start happening, and then finally, you know, we are going to commit to one single event that is going to be pursued aggressively as the commercial candidate after all that testing that has been done. But one question remains, and is, okay, so how is that go ahead and set up a research objective? And so that research objective normally comes, if you remember, I mentioned we are representing things linearly in terms of our trade development process. But there is actually a feedback loop into research organizations. And so those product concepts that we refer to and that we use to base our objectives on, they come from what are the needs that the consumer, or in this case the customer, has in the farm. And so we are constantly scouting for, okay, what are the resistances that are developed? What are the dynamics of the pests that we are trying to address? And so try to bring that back into the discovery program to make sure that those needs are constantly revised and maintaining context. And so there are also technology needs. So associated with a product concept, there are technology needs. As new technologies get developed and we get to apply them, we sometimes have to develop new technical platforms. One example is the case of CRISPRs. Technologies that have been incorporated into our platform to be used in different ways, and then we define what is the best use of the technology and for what purposes, and how does it fit the models that we have. Very importantly is the fact that product concepts are jointly developed with our commercial R&D units, and this is something that many scientists say that probably they didn't sign for that. But that's a reality that we need to make sure that what we are going to tackle and the investments we are going to make in terms of our research platform always get aligned to a benefit not only for the farmer but for the organization as well. As you saw in one of the presentations today, it's a very large commitment of money and time into these pipelines, and so they have to make sense. Is a good, personally I think it's an excellent compromise within an organization to have those objectives jointly developed. And then these new discoveries, these new traits must have some technical attributes that we cannot get away from. And those have to do with the insecticidal potency. More and more, I come to realize that insecticidal potency is not necessarily the biggest obstacle. I think you can get to that very easily. The problem is that insecticidal potency comes with a cost. And so normally when you have that potency, you have an effect on the plant. And so that is where you need to start figuring out, okay, so do I, what is the potency level that I need in my trait that is not going to compromise my crop? The other aspect is spectrum. So sometimes you have solutions that are highly potent for a given pest, but only covers one pest and one pest only, and so that doesn't really work for what you really need to do. So very often we put as a threshold the minimum number of pests that a solution has to provide solution for is 2 or 3, depending on how you are configuring your trait platform. So pest spectrum is an important thing. And then the other element is the fact that we need to have new modes of actions. And new modes of actions, you heard referred to it in multiple presentations today. That is, and during the discussions, that is an important element. In the case, this is in particular for the insect control traits, but new modes of actions get you away from the resistant pests. And so that's something that you always want to do. You want to continuously rotate those modes of actions because you don't want the constant pressure with the same mode of action, and the pests are going to circumvent that. That certainly has been broadly exemplified today with the case of herbicides. And in the case of herbicides, there hasn't been any mode of action in the last 30 years. And so obviously the problems we are seeing today are reflective of that particular situation. And not only that, but the herbicide tolerant solutions on the trade side, normally have come in some cases 30, 40 years after the chemistry was applied for the first time in the market, in the farm. So it has been plenty of time for that selection to occur, and also, as you heard today, with not such a good stewardship and integrated pest management systems. So it's all these very complex, and some of these things also apply to the case of insect control. And so what I have there are two of the pest classes, pest groups that we work the most with. And so those are the Lepidopteran pests and the Coleopteran pests. And so those are areas, one, we call it, in our internal lingo, we call it, the Leps are above ground pests, and the Coleopterans are our below ground pests. So that's how we refer to them. So you probably will see some of that, or have seen some of that in the slides already. So talking about new modes of actions, so I wanna start the next part of the talk with kind of offering a perspective. This is the picture of all the insect control traits that have been put out, and I decided to put in there the name of the genes. So most of those genes, except for one, are all coming from Bacillus thuringiensis. So this has been a source, a gene source that has been broadly used and highly successful as well. So there are constant and different modes of actions that have been extracted from it. Moreover, I'm not saying that this is not valid still. So this is still a very valid approach, but there are also other technologies that are being applied to this. Protein engineering technologies, gene shuffling, all these other different technologies are being used currently, as well as the discovery of new bacillus strains to mine for the identification of new genes. But what you see here is that all of these genes here are Bt genes, except for this one. This is an RNAi technology that has been developed by Monsanto, and so this is a gene, an antisense, an RNAi, not antisense, an RNAi technology that has been developed against the SNF7 gene from Diabotrica virgifera. And so this gene is a gene that is involved in vesicle trafficking in the insect. And so what has been done with this technology is that when the insect eats it, so it's going to interfere with trafficking in the cell, and so then it makes the insect sick and eventually dies. But other than that, all the other technologies have been bacillus derived. So this is just to illustrate how the industry now is starting to see this. Obviously, we know at some point the well is going to go dry, and in terms of sources of genes, and we need to start looking for new solutions in other sources. So what we are doing in my group? So what we are looking at is try to create as much diversity in the screenings that we are doing as possible. And so we are using not only bacillus, where we have a large collection of strains and we are still mining those, but we are also looking into non-BT bacteria, where we have other types of isolates. Some of those, I'm going to have an example of one of those. And then we are looking into plant-derived proteins. We are exploring that space as well, making sure that we use plant sources as well in the crops. That's an idea that is very attractive and appealing. And then also we have the protein evolution technologies that I was referring to, like protein engineering, shuffling, and mutagenesis as technologies. And then we are looking into whole biomes as well. So samples where let's say we look at a micro ecosystem of that is of interest to us for some reason, being that dead insects, being that whatever, a patch of soil where we see plants that are surviving well while others get eaten, all sorts of different sources like that. So we are looking at the biome space as well, trying to make sure that we explore as many possibilities as possible. And now with the ability that we have to sequence and next generation sequencing is relatively straightforward to do it and go and mine all that space. So sources are one thing, but then you have to take different approaches as well. And I brought up this slide because I thought that it can kind of give a general idea of the approaches. This is part of our staging in what we call stage 0 discovery. So this is what we go through normally. And so we take different types of approaches. We go from taking an activity approach where we basically what we are looking for is in the extracts of the organisms that are our target, we try to identify, actives or activity against a given pest. And so for that, we characterize if that is a small molecule or if that is a protein. Obviously, we are interested in proteins, so we try to select for those. We also can take a genome approach, like what I described to you, sequencing, exploring the genome of a potential good source of genes. Or we can take the biome approach, where we use a metagenome-based approach. We don't necessarily look for where is exactly the gene coming from at the beginning. We will do that later, but we just simply take a broader approach with that technology. And we have other things that are, I call specialty, because we take sometimes a one-off type of approach to go and pursue a very specific pest or something that is, let's say, something interesting that we have identified in academia or that we are bringing from a collaborator and we are testing in very specific ways. Then after those approaches get taken, we are obviously identifying some of those actives. One of the big elements is to test properly against the target pest. It's a big arrow here, but not as big as it is supposed to be, because this is probably the bulk of the work that we do in trying to select and trying to identify good ways to select. Your discovery platform is as good as this is. I mean, if you are testing properly, it's like in anything that you can do in any lab, in any research program. It's only as good as your screenings are. And then obviously we go and select those that offer us the phenotype of interest. And so here you see, this is something that you probably are very familiar with. I'm pretty sure you are very familiar with. All the different technologies that we apply or the different areas that come into play for either our gene sourcing, the identification of candidates, what we call actives. Actives are when they already have proven an activity in a crop of interest against a pest of interest, and also the lead generation, which is when we are already moving heavily into the testing in the crop. And so now getting into more specific examples. This is now towards the end of my talk. Gonna cover very briefly some examples that we have, and this is coming from the non-BT bacterial screening approach. And this is a gene that we have called IPD72. And this is a gene that is coming from Pseudomonas chlororaphis, and this gene is testing very well in corn. In this case, it's a below ground corn rootworm target. And so what you see here is the type of approach. You don't know why it's covered here. There was no intention to cover anything there. But what we are doing is really testing against identifying as many homologs of the gene as we can in our collections to make sure that we mine all that space. Once we identify one of the actives, we try to go and mine and identify as many similar of those of that particular gene or protein as we can, and then we test them as well. So we try to be comprehensive with our testing program. So what you see here is this is a commercial tester. This is something that is already in the marketplace that is effective against corn rootworm. This is a negative control. This is an untransformed, non-transgenic set of plants. And then you see 3 events. These are commercial grade events that are being tested against, 5 events that are tested against, that the pest is tested against. And you see what the difference is when you have a susceptible one and when you have the gene in the plant. So the plant gets quite well protected from corn rootworm. The other thing that you see here is this is part of what we do. This is an interesting and important part of what we do, which is for some of these commercial controls, for some of those commercial genes, there may be areas and locations where resistances may have been already developed. And so what we do sometimes is, since that we know that is possible, we have stocks of some of those resistant or tolerant lines, insect lines. And so we go and test the new genes against those resistant corn rootworms. And so what you see here is this is the negative control. This is the susceptible corn root insect, corn rootworm. And so you see it being tested against 2 of our events and 1 of the commercial gene technologies that you saw listed in that timeline that I showed you. And here is what happens when you test against the already resistant insect lineage. The commercial control here is susceptible, is proving susceptibility. This is injury, okay? And you see the new events that we are generating with this IPD72 are showing very low level of injury. And so that's how we continuously test against what is happening already in the fields. The other example that I wanted to show you is a gene that we are very excited about. This is something that we call IPD79. This is a gene that is coming from a fern. We have identified some ferns as very good sources of some of these genes. It's very rarely that these ferns from where we are taking these genes show any injury, even in the field. You go to the forest and they hardly have any bites from any insects. And then we derive a homolog. This was selected in our screens as well from another type of fern. And you see we have tested both of those already in our program, and they show an excellent resistance. And here in this graph, what you see is this is IPD72, the one that I presented in the previous slide, against the same corn rootworm. And you see the level of injury here is very low for that. The same thing happens for these 3 events that you see here that are derived from the fern. And this is the commercial check. You know, this is all in the susceptible corn rootworms. And you see how the negative control has a high level of injury, and this is the picture that corresponds to that. So in other words, so now in this case we have tested this against the western and northern. We haven't tested yet against the Mexican corn rootworm. That's another one of the ones that we target, but it's very important to us that there is a good degree of control of the western and northern corn rootworms. So the point here being that, you know, we are trying to resource to as many of those diverse sources as possible. So we are trying to diverge our program to have more than just BT-derived genes in our discovery pipeline. And so so it was much said this morning about herbicides, and this is one of the herbicide technologies that we have. And so I'm going to use this to close my talk. because I think it's important to, you see how all these different things and the science gets done and how these platforms work, but at the end, there is also something else that has to happen in order for these things to work. So here what you see is two herbicide tolerance genes. There are two types of chemistries that work with Enlist. One is 2,4-D and the other one is quizalofop. And so the 2,4-D is used with soybeans and the quizalofop is, the fop family is used with corn. And so we have the crop, the herbicide tolerant crop that we have generated have either the AD12 that is coming from Delphia acidivorans, or it comes from the AD1 which is coming from Sphingobium herbicidivorans. So those are the sources for that. And so this work started more than a decade ago. And we are now in the process of commercializing more and more versions of these things. But this is how it looks at the end. And so there's a whole platform that has been developed, and it's not only about the trait, and it's not only about the chemistry, and I'm going to try to explain that in a few words here. So what you see here, this is the model of how what we call the Enlist weed control solution looks like. So there's a component on traits, and here you see represented 3 different crops, corn, soybeans, and cotton. This concept here is Conquesta, which is something where we are deploying in Brazil. It's among the better pyramid soybean trait packages where we have insect control and herbicide tolerance all together for the Brazil market primarily. And then we have here, you see there are 2 solutions for soybeans. Soybean is really one of the drivers as a crop that we have for herbicide tolerance. And you see this is called Enlist E3. There are 3 modes of actions that are stacked on this, and we have developed this as a single molecular stack. And so this is tolerance to 2,4-D, to glyphosate and glufosinate all together. And then there is this management resource program that we created also, and this has to do with all what I was saying at the beginning. You have to be responsible with the products that you are releasing, because there is a stewardship component that comes with it. And this is benefit for everybody. This is a benefit from regulatory agencies. This is benefit for the farmer, because this helps control development of resistances. And this is obviously benefit for the organizations that are producing this, for the industry as well, because this results in trade durability. And then there is obviously the chemistry component here. And so there is a solution that has been developed that is called Enlist Duo. I want to take a couple of sentences to explain this. And so this Enlist Duo is a mixture of glyphosate and 2,4-D for the purpose of the herbicides. So that's the mixture. But there is a component there that where we spend a lot of time and resource making sure that that formulation was going to be right. And so that's technology that we call Collext. And that Collext technology is what we have put into this product that allows for less volatility and drift. And so this has turned out to be a great benefit for everybody, because you have heard the news about the recent problems that there have been. But paying close attention to integrate properly your platform pays off. And here is where you integrate the whole program, and then it results in really a big benefit for everybody. So there's a few bullets there summarizing. So it's obviously developing solutions requires understanding of the customer needs, requires that we have clear objectives, that there is a benefit in safety for the customer and consumer and the environment. All those things will pay off in the long term. We follow in industry. We follow what we call the trade development process. Yes, it's a disciplined approach. and it's milestone based. So it always goes hand in hand. You gotta do all what you gotta do, and you have to be very milestone based. It's a process that takes between 10 and 18 years. It's a long time to go from beginning to end. And then a pitch for insect control and herbicide tolerance traits. I personally think that they are key in increasing productivity per acre. It's going to be one of the key things that we do in terms of increasing productivity. There is a great customer demand for these type of traits. I don't see that getting any lower. And then there is certainly a lot of complexity in developing these traits. There's a lot of complexity in the pest dynamic, pest populations, and so on. And it's really what we need to do, because there is there are big demands from a growing population, and land is not increasing. So with that, I just want to say thank you again for the invitation. It has been great to be here and learn from others. This has been a wonderful experience. So if you have any questions, happy to answer. It's now time for dinner, huh? I think there was one question.
You going? Yeah. Go for it. You'll beat him.
Rodrigo, very nice. And you mentioned the plant growth trade-off, and especially with the insect work. I wondered if the new generation is overcoming that problem, if that's typical of hygiene, or if that's something that would be better solved.
Yeah, good point. So yeah, that's one of the things that we are trying to overcome. Phyto problems in the plant are the type of things that we are trying to avoid with BTs. BTs are highly potent. With BTs, we don't have any issue with potency, really. The issue is to select for that compromise that I was talking about between potency and good plant characteristics. But yeah, that's part of the reasons why we are going into different sources. Definitely. Yeah, it's a driver.
Thank you.
Good. Thank you.
So we've reached the end of the day, so I thank you guys, those of you especially that stuck with us all day. We appreciate it. It's a lot, but we really hope that you guys are able to take from it and really, think about the future and everything, from looking at a fern and thinking, why don't bugs attack it? Let's find out, right? And that's probably what I'm taking away from today. So first of all, I'd like to thank you guys again just for attending. We really appreciate it. We do it for you guys. We have a couple students here that traveled, so thank you very much. Our speakers, thank you guys so much for giving us the day. Some of them came in last night to meet and to socialize with us, so thank you so much for that. The crop science department, they've been big supporters. They've not only provided some money, but also they're not here, but a lot of the office staff has really helped us do things like book flights and hotels and the logistics of food and everything. We really just couldn't make this day happen without that. The Office of Research, Dr. Blair was here earlier, but they provided funds as well too to support this. The committee, I think some of them left to go clean up, but if you're still here and you're on the committee, can you just stand up? Yeah. I'm up here speaking in front of you guys right now, but these guys really are the ones who put this thing together. Incredibly hard work. We spent about a year planning it. Pretty much only two of us that helped plan it last year continued on, so a whole fresh new batch that came on with new enthusiasm, new energy to plan this year. So it's an incredible job. So finally, just like to invite you guys to come out to Crane Alley. It's on Main Street in Urbana. 6 o'clock we'll have drink tickets there, so you can get some drinks and some food and come either talk about science or don't talk about science in an informal setting. So thank you very much. Finally, if all the speakers we could just meet real quickly, and the committee members get a picture after this. Maybe we can meet just upstairs. That would be amazing. Thank you.